in 15 minutes. Extended release is for prolonged duration of action. So there will be 3 time points, like in 1 hour it should not release more than 20%, in 4 hour release should be 30 to 50%, in 10 hour or 12 hour not less than 80%. Like this, like that criteria will be there. So those are the responses. So now we have seen in the previous slide there are so many designs which can be used for optimization studies. So how we can select what design we have to take, right? It is not like that for every optimization we can use the factorial design or central composite design or any design. That depends on number of factors. So for example, if we are doing optimization for formulation, all design can be used. If we are using process variable, then SMD can't be used, simplex mixture design. Number of factors, if number of factors are less than 3 or equal to 3, we can use any design. If it is 4 to 6, we cannot use simplex mixture design. If it is more than 6, then we have to go for fractional factorial design or Taguchi design. So basically if the factors are more, right? So for example, if factors are more than 6, so what we have to do? We will select, first we will do the screening. Screening means out of these 6 factors, which are the 2 most influencing factors which are affecting our response. Okay, so if factors are more, first we have to go for screening to reduce the number of factors, because unnecessarily our designing will be complicated if we are using more factors. So first we will select what are the most influencing factors that may affect the response. Factor level means we have discussed in the previous slide. Levels means the quantitative term which we are giving to any factor, like 10 mg, 20 mg, 40 mg, or type of disintegrant or type of binder. So those are levels of the factor. Linear model means where there is no interaction between the factors. For example, if you are using disintegrant and binder, those are behaving linearly. Linearly means if you are increasing the disintegrant, disintegration time is reducing. If you are increasing the binder, then disintegration time is increasing. So according to the quantity of these excipient, the results are coming. But sometimes it may happen that you are increasing, then also it is not increasing proportionally, right? So in those cases we have to study the interaction model, where factors are having interaction between them. They cannot behave independently, or they cannot give the results independently. In those cases we have to go for interaction model or quadratic models. Next please. So overall plan for optimization, first we will selection of excipient, selection of process, what are the excipient, what is the process, identification of independent variable. Independent variable means response, right? Most influential variables, here influential variables means factors. Next, factor level, response variable, identification of model or experimental design. In the previous slide we have seen the table. By using that table we can select with which experimental design to be used. Formulation and evaluation of drug delivery system. Some formulations we have to make before going for further optimization. Statistical analysis, by using software we have to go for statistical analysis, mathematical model, response surface methodology, and search for optimum targets. So first once we are having the response surface graph, we can select some of the formulation, right? So which are giving results as per our requirement. Then verification of new experiment. Verification of new experiment means whatever response surface graphs we are getting, from those we will select some of the formulation, and then practically we will make the formulation and we will check what are the theoretical results and what are the practical results we are getting. So if the results are almost same, then we can consider that whatever optimization we have done, whatever equation we have generated, that is okay. Next slide. Next slide. So when the number of factors are more, we can go for either fractional factorial design or we can do screening of factors, right? Next. 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 So for example, here we have selected 3 factors, right? First is magnesium citrate. Magnesium citrate is used as a lubricant, right? Lubricant in the formulation. So it is hydrophobic in nature, so it may retard the dissolution or disintegration of the formulation. Next is drug and next is starch. It is a binder, right? So levels we have given, low and high levels. So we want to study at the different levels. Next. Previous slide, please. So here what we have done, we have selected 3 factors, and what are the number of levels? 2. So whenever we are giving, so in short form we can consider as LF. So L will be in the bottom and F is a superscript. So levels are 2 and factors are 3. So 2 to the power of 3. So 2 into 2 into 2, it will become 8. So number of formulations will be 8. So if we are selecting 2 levels and 3 factors, number of formulation will be 8. So these are the formulation. Minus sign denotes the low level. Whatever we have seen in the previous slide, there were 2 levels. One is low level and another one is high level. So minus is low level, plus is high level. So these are number of combination. Number of combinations means these are the experimental designs. These are the formulations which we have to manufacture. 1 A, B, AB, C, AC, BC, ABC. So these are 8 formulations which we will manufacture, and response is thickness or disintegration time, whatever we want, we can select the response. Now we will see the case study so that it will be clear how we are doing the optimization. So this case study is for formulation of nanoparticles by desolvation method, and where the factors were more. So we have done screening of factors by using Taguchi design, where we have reduced the number of factors. After getting 2 factors, after screening we have got 2 factors, where we have done further optimization by using central composite design. Next please. So the plan of work was model generation, response surface analysis, generation of polynomial equation, which we will see in subsequent slides, and selection of validation formulation. Selection of validation formulation means theoretically we will select the formulation and then we will manufacture it practically, and then we will compare the results, what are theoretical results and practically what is the result we are getting, so that we can be sure whether whatever optimization we have done, that is okay or not. Next please. So evaluation parameter or responses. So here responses were particle size. As we are manufacturing nanoparticles, particle size is important. Particle size distribution. Okay. So next, entrapment efficiency, drug loading, drug release study, and mucoadhesive strength, as these are gastroretentive mucoadhesive nanoparticles which should adhere to the gastric mucosa and release the drug in sustained release form. Next please. Next. So here what we have done, we have dissolved 200 mg of drug in water and then transferred it to gelatin solution. Further, chloronic F68. Chloronic F68 is a surfactant which was added and pH adjustment done. Magnetic stirring followed by acetone as a desolvating agent. So what acetone we are using as an anti-solvent. So it will result in the formation of nanoparticles. Nanoparticle suspension will further we will add glutaraldehyde. Glutaraldehyde is a cross-linking agent which will prevent the release of drug from the nanoparticles when it is in the formulation form. Okay, inside the body it will release, but when it is in the formulation form it will prevent the leaching of the drug. After 6 hours of centrifugation, nanoparticle precipitates were formed and further freeze drying were done to get a lyophilized product. So here factor work, amount of gelatin. Gelatin is a polymer. So two levels we have taken. Low level is 200 mg and high level is 1000 mg. Amount of glutaraldehyde 0.1 and 0.5 mg. Amount of chloronic F68 50 and 250 mg. Acetone addition rate 1 ml per minute or 5 ml per minute. pH 2 or 4. Stirring time 1 hour or 5 hours. Stirring speed 200 and 1000 rpm. So these are the 7 factors. So we have done screening. Next please. So these are the formulation. By using the Taguchi design, only 8 formulations were made for screening of these 7 variables. So minus denotes low level, plus denotes high level of the factor. Next please. So these are the actual value which were assigned to levels. Next. So after formulation, these are the results which we have got for 8 formulations. Okay, so particle size was ranging in 165 to 1610 micron. PDI is particle size distribution, that is polydispersity index. So minimum value denotes that our particle size are uniform. If the PDI is more than the particle size are scattered, means it is not uniform. So PDI should be as minimum as possible. Next, entrapment efficiency. We should get highest entrapment efficiency. Drug loading T60 is amount of drug time taken for 60% of release. Q6 is amount of drug released in 6 hours, and MS is mucoadhesive strength. That is done by using texture analyzer equipment. Next. So after getting those results, we will enter those results in Design Expert software, and after doing ANOVA analysis, we will get these coefficient values, and right side is a Pareto chart. So these bars denote its impact. So the bars which are having the more length when compared to other factors means those are the most influencing variable which are affecting our response. Okay, so out of these 7 variables, we found that gelatin and chloronic F68 are having the maximum length of the bar, means these are the variables which were affecting the response when compared to other variables. Next please. So then gelatin and chloronic F68 were further used for optimization study, where we have used 3 levels. So 200, 600 and 1000 mg for gelatin, and 100, 200, 300 for chloronic F68. Here we have used the central composite design. Next please. So in central composite design, formulations are 9, and you can see that CF10, CF11, CF12 and CF13, those are same. Okay, so here 0 means, what is 0? 0 is the mid value. Okay, in the previous slide, please go to previous slide. Here what is low, intermediate, high? 3 levels we have selected. So for gelatin, 200 is low, 600 is intermediate, 1000 is high. So for middle value we are assigning 0, for low value we are assigning -1, for high value we are assigning +1. Next please. So 9 formulations were there, but we can see that C10 to C13, those are same formulation. All are having 0, 0 assigned value, right? So what are those? So those are center point. Center point means deliberately we are adding the experiment to check the precision of our experiment. Whatever experiment we are designing, whether it is giving precise results or not. Means if we are preparing formulation with the same condition, means 0, 0. So it should give the same result, right? If we are taking only one formulation, then we should not come to know that whether whatever we are designing that is reproducible or not. So if we are making 5 formulation with same composition, and if those are giving same results, then we can say that whatever we are doing that is giving reproducible results, right? So for example, disintegration time is 5 minutes. So every time it is giving 5 minutes or plus minus 5-10 seconds. So it is reproducible. But if with the same formulation, same factor, same manufacturing technique, if once we are getting 3 minutes, next time we are getting 8 minutes, next time we are getting 5 minutes, means reproducibility is not there. Then there is no use of doing these optimization or any study. Whatever we are doing, it should give reproducible result, right? So that's why we are adding the center point formulation C10, C11, C12, C13, which is similar to C5. Right, in C5 also there is a assigned value of 0, 0. So 4 center point formulations we have added to check the reproducibility of result. Whatever result we are getting, those are reproducible, not by chance. It is by design. Next please. So these are 13 formulation and values assigned to those levels. Next. So these are the result for 13 formulation. What is the particle size, PDI, entrapment efficiency, drug loading, T60, Q6 and mucoadhesive strength. So after doing ANOVA by using Design Expert software, okay, so you can also download the Design Expert software, but it will be a 30 days trial. After that you can't use. So by doing ANOVA in Design Expert software, by using central composite design, we will get these coefficient values and p values. Next please. So this is a model generation. This is a polynomial equation, right? So here we can see B0 is the intercept, B1 x1, B2 x2, B3 x1 x2, right? So B1, B2, B3, these are factors, and x1, x2, sorry, B1, B2 are response, and these x1, x2, x1, x2, these are factors. So when we are writing x1, x2 means there is an interaction between those terms, right? When there is only one term x1 means independently we are seeing the result impact of x1. When there is only x2 means we are seeing result of only x2. When x1, x2 are there means there is an interaction between those two terms. Okay, so then we can get the equation, polynomial equation for each of the response. So response were PS is particle size, PDI is polydispersity index, entrapment efficiency, drug loading efficiency, T60, Q6 and mucoadhesive strength. Okay. One second, please, in the previous slide. So by using these equation, we can design a formulation to get the expected result. So without practically making a formulation in a lab, we can just enter the value of x1 and x2, right? So x1, x2 values we have seen in the previous slides. So those values if we are doing here and if we are calculating, we will get what will be the particle size of our formulation. So without making actual formulation in your lab, by using this equation only you can get the desired result. Whatever is your desired result we can get by using these equation, whether it is for particle size, PDI, entrapment efficiency or any response. Okay, next. So these are the response surface plots. So these are 3 plots where response surface plot, 3D plots, where we can, some clarity is not there. We can get, means what is the impact of the levels of factor, means if the factor level is increasing, the response is decreasing or it is increasing, we can get to know from this response surface plot. Sometimes it is linear with increase in the quantity, response is increased. Sometimes you can see the curvature. Once it is increasing and then it is decreasing. Sometimes it is decreasing and then increasing. Means there is an interaction between these two factors. Therefore, we are not getting linear graphs, right? Next, please. So by using those equations, we have seen in the previous slide that equation, polynomial equation we have seen. By using that equation, we can make a table in simple Excel sheet, and we can go for feasibility and grid search. Feasibility search is a somewhat broader selection. Means, for example, here x1 and x2 we are taking, right? So x1 is given in left side and x2 in the top. So x2 ranges from -1 to +1 and x1 ranges from -1 to +1. And here we can see that particle size, our selection criteria was particle size minimum 191, maximum 475. So which is the area in this table where we are getting our desired response. So that area is where x1 is 0 to 0.4 and x2 is 0.6 to 1. Okay. So these are the levels. Next. Similarly, for other factors also, we will be having our desired range. Drug loading, what is our requirement? T60, what is our requirement? So based on that, we will select the region for feasibility search. Next. Can you please go to previous slide? So here we can see x2 is 0.6 to 1 and x1 is 0 to 0.4. So further we will go for grid search. Means this area we will enlarge. What is 0.6 to 1? That we will enlarge in next slide. Here you can see x2 is 0.6 to 1, right? Further we have subdivided it, and x1 is 0 to 0.4. So here also we will be having our selection criteria. In the previous slide we have seen that our selection criteria was with minimum and maximum value. That particle size minimum should be this much and maximum should be this much. But for feasibility search, grid search, we are giving a specific value of particle size less than 300 micron, PDI less than 0.3. We know that minimum PDI is better. If the PDI is less, means our nanoparticle particle size are uniform. Okay. Entrapment efficiency more than 65, 100% you will never get, right? So whatever maximum is possible, that we have to select. Drug loading is 80%, P60 is 14 hours, Q6 is 30%, mucoadhesive is strength is 4.83 gram. So if the mucoadhesive strength is more, means in weight it is more, then mucoadhesive strength is more. If mucoadhesive strength is more, means nanoparticle will adhere to the lining of the gastric mucosa for longer period of time, and it will release the drug in a controlled manner, right? Next. So this is a grid search for different formulations. Next please. Next. Previous slide please. So from the grid search, whatever is the green selected cells are there, means we are selecting the formulation, right? We have not manufactured these formulation, but by using equation we are selecting 4 formulations from intensive grid search. Okay. So whatever green cells are there, formulation we have selected. For example, 31.8 is given, right? So in that case, for that x1 is 0 and x2 is 0.64. So level is for 31.8, level of x1 is 0, level of x2 is 0.64, right? So like this, we have selected 4 formulations from this grid search. Next slide please. This is the overlay plot. Here you can see that a yellow region in the plot, right? So whatever is our desired criteria, we have given in the table, that particle size 200 to 250, polydispersity index 0.24 to 0.36. Like that we have given our criteria. So if you will make your criteria more stringent, then this yellow region will be, what will happen? it will become small. Means chances of your formulation within that region will be less. If you are making it broad, yellow region is broad, means number of formulations can be more. So in the software, if you are clicking just in the yellow region, if you are pointing with your mouse and clicking it, then you will get the formulation. Like we have selected one formulation where you will be having x1 and x2 values, means factor, and as well as response. So by using that overlay plot without manufacturing any batch, in the yellow region we can point anywhere and we will get the combination. What is the concentration of x1 and x2 and what is the response that we will get. Okay. So without doing trial in lab, by only using computer system, you can have the formulation combination, what is the composition of the formulation and what will be the desired result. So from here we have selected one formulation and from that grid search we have selected 4 formulation. If we want, we can select 10 formulation from here also. Whatever number of formulation you want, you can select. And that yellow region is your desired region, right? So that desired region is based on the constraints level which we have given in the table. For example, particle size 200 to 250 micron. Likewise for other factors. Okay. So total how many formulations we have selected? 5 formulations we have selected. 4 formulations from grid search and 1 formulation from overlay plot. Next slide please. So validation of the optimization analysis. Validation means whatever we have done, we need to validate whether it is right or wrong. So for validation, what we will do? We will prepare the formulation. Whatever we have selected from the computer system, 5 formulation we have selected. Practically we will manufacture those 5 formulation and we will see the results. Next please. So these are the 5 formulation which we have selected. V1 is validation 1, validation 2, like 5 formulation we have selected. V1 to V4 from that grid search and V5 from overlay plot. So those are the coded values which we can get from those graph and table. 0, so 0 represent what quantity, so that quantity we have mentioned here. Next slide please. So after making formulation, so predicted value is the value which was given in the table or that overlay plot. Means x1, x2, which if you will take this much, response will be this much. So those are the predicted value, and experimental values are the values which we received after making a formulation. So you can see that predicted value was 258.8 nanometer and experimental value is 258.4 nanometer. So difference, residual is difference. So difference is only 0.4 nanometer. It is very less, right? So likewise you can see for all the formulations. So the results are matching with the predicted value. We can see from mean of percent error. It is only 0.129. Means error is less than 1%. Means we can say that whatever designing we have done by using design expert software, we are getting the desired results, right? So next time no need to again go for this validation. Means we are doing checking whatever equation we have generated, whatever overlay plot we have made, whether it is giving good results or not. And when we have done successfully validation, it is giving good results, mean next time need not to formulate any batch in the lab. Directly by using that equation or overlay plot, we can select the factors level and we can get the desired responses. Okay. Next please. So advantages of systematic optimization technique. It requires fewer experiment, right? Like we have done first screening and then we have done optimization studies. So for 7 factors. Initially factors were 7. Then after screening we got 2 factors. Further, those 2 factors were optimized by using central composite design. By using that, we have generated the equation and that polynomial equation and overlay plot, right? So next time, if you want to get any desired result, for example, if you are going to your boss or going to someone, if you are presenting that I have got 250 nanometer of particles for my formulation, whatever I have manufactured. He will say, no, I want 200 micron or I want 300 micron. Then again, if you are not doing systematic optimization, means again you have to go to lab, again you have to do so many trials, and then you will be able to get the desired results. But if you are having the equation, if you are having the overlay plot, then need not to do anything. Just you will select that equation, or you, by using that graph, you can select the concentration of your factors, right? X1, X2. What is the quantity of those factors? You will select, so that you will get 250 nanometer of nanoparticles. Likewise for any other factor or in any other response, right? So for immediate release formulation, if your release is 75% in 15 minutes, but someone says no, I want 85% in 15 minutes. So need not to do again so many trials. Just you will go to equation and polynomial equation and graph, and you can select and you can proceed, right? So require fewer experiment, can trace and rectify a problem in an easier manner, yield the best solution in presence of competing objectives. Means if there is any interference, so we can get the best solution. Can change the formulation ingredient or process independently, right? So we can change the process independently and formulation variable independently, and without making a formulation we will be able to know that what will be the response, right? So if you are increasing or decreasing the level, what will be the response you can get without manufacturing a batch. Save a significant amount of time, material, cost, all those things will be saved. Can predict the performance of formulation even without preparing them. So no need to formulate in the lab. Detect and estimate the possible interaction among the variables, like we have seen in the 3D graph, right? Sometimes it is linearly increasing, sometimes it is increasing and then decreasing, or sometimes it is decreasing and then increasing. So if it is not linear, then there is an interaction between the vectors. So by seeing graph only we can come to know whether there is any interaction or it is behaving linearly. Okay. So thank you. If you are having any questions, you can ask. So those questions may be related to this presentation, or anything related to research and development, or related to formulation R&D, or any question is welcome. Okay. So remember in mind that question is never wrong, answer may be wrong. Okay. So maybe I may not be able to answer, but I will try to answer. If I am not able to answer, then definitely I will tell the answer later. So you can ask any question. Related to optimization, formulation designing, factors, or research and development, or related to industry, or related to interview, that after completing your B form, what will be the possible questions, or after completing M form, if in particular field you want to vote, what will be the questions. Anything you can ask. I will be happy, as many as questions you will ask, I will be more happy.
So she has a question.
Yes, please.
Good morning, sir. I want to thank you for your insightful presentation.
Thank you.
I have a question regarding Taguchi design.
Okay.
We are using Taguchi design in various fields, right? But in pharma field, why we are using Taguchi design? Is it to improve the quality, or to decrease the time and to decrease the cost?
So Taguchi design, normally initially I told that factorial design, right? I told LF. L is level and F is factor. So if you will see, there were 7 factors, right? In our presentation, there were 7 factors and 2 levels, right? So 2 levels mean 2 raised to 7. If we will go to factorial design, then what will be the formula? 2 raised to 7 means 2 into 2 into 2 into 2, 7 times you have to do. So what is the answer exactly? I don't know. Maybe number of experiment will be nearly 100 or 90. So if we are going with the Factorial design levels are 2, right? Quant means low level and high level, and factors were 7. So the formula will be 2 raised to 7, means 2 into 2 into 2, 7 times we have to do, right? So those will be nearly 100 or whatever it is. So number of formulations is very much. So in that case it is difficult to formulate 100 formulations, means it is, anyone will say better you go with one variable at a time or one factor at a time. So Taguchi design, why we are using? To reduce the number of experiment. So in Taguchi design we have seen that we have manufactured only 8 formulations. 8 formulations we have done, and we were able to screen out of 7 factors, we were able to screen out what are the most influencing factors. So only 2 factors we got. Then further for optimization study, what we have done? Central composite design. In that we have selected only 9 formulation. 4 formulation was center point formulation, right? So 9 plus 5, 14, 14 plus 8. By using only 22 formulations, we were able to validate our optimization technique, where factors were 7 factors. For 7 factors, by doing only 22 experiment, we were able to optimize our formulation. And next time onwards, no need to manufacture a batch in the lab scale. Just you can use that polynomial equation or graph, and you can have the desired result. Whatever is your desired results, accordingly you can use x1 and x2, your factors, means amount of gelatin, amount of chloronic x68. In this formula, in this example, if you are doing any other formulation, you are having other factors, other responses, so accordingly you can design your formulation, right? So here in short, Taguchi design we are using for screening of the factors. So to reduce the number of factors, because it is difficult. If anyone will say that 7 factors at a time you have to study, it will be difficult. So what we will do? What are the 2 most influencing factors that are affecting our response? Those only we will study. So here we are using that for screening purpose. Okay. Thank you.
Any more questions? Students, please do not hesitate. Anybody else?
You can ask any question, right? It is not like that you have to ask only related to optimization.
So by using polynomial equation, we will make the formulation.
Right.
Desired formulation by
Right.
taking design of experiment.
Right.
So we are having a challenge with respect to the stability of the formulation.
Right.
So do we have any such kind of design of experiment to improve?
Right. Means in that case, your response will be stability. In stability, what you will do? In stability, either you will check the assay or related substances or drug release, means whatever are your CQA, critical quality attributes, right? So whatever your CQAs are in the formulation. So in the formulation, normally what we are checking in stability study, why we are doing? So whatever formulation we are making, our main aim should be, it should be effective as well as stable, right? If it is not stable, then it is of no use. If it is stable but it is not effective, then also it is of no use. So it should be effective and stable. So in case of stability, our response will be SA, right? SA means 100%, it should be near to 100%. Range will be 95 to 105. RS means related substances, impurity. What is the impurity which you are getting in the stability? Okay, dissolution profile, whatever initial dissolution you are having, whether you are getting same or not. So that also we will check during stability, because initially whatever bio studies we are doing on human beings, right, healthy volunteers, at that particular point of time, if you are having the fast release, but upon stability, if the release is getting decreased, then it is of no use, because the formulation was effective in the human volunteer when it was releasing fast, but upon stability, if it is decreasing, means during the shelf life, release will be decreased, means it will become less effective, right? So in that case, so for our study, our responses will be assay related substances. For assay, what will be our criteria? It should be between 95 to 105, or we can make more stringent, 98 to 102, right? RS, related substance, it should be minimum. Whatever minimum is possible, that is best, right? So our response will be minimum RS. Water by Kf, it should be minimum. Dissolution, whatever initial criteria you have set for dissolution at the point of release of formulation, means whatever initial it is showing dissolution, same should be criteria for stability. You cannot relax that criteria. Means for example, initially if you are giving 15%, 80, not less than 85% in 15 minutes. So in stability also it should release not less than 85% in 15 minutes. Then only your formulation is stable. So in case of optimization, for responses, in place of your whatever you have selected for initial, same will be for stability. Okay.
How can we define these specifications?
Specification means for every product, means for impurity. For impurity, we will look into ICH guidelines. Okay. So anyone can say the full form ICH? Full form of ICH.
International concepts of harmonization.
Of?
Harmonization.
हाँ आगे भी है. Harmonization of technical requirement for pharmaceutical products for human use. So that is the full form of ICH. No issues. So it is an agency, right? So it gives guidelines for impurity. What will be the limit of impurity? And if you are manufacturing a official product means that is given in the monograph, USP, BP, IP. So in those monograph there will be specification for each and everything. Assay, how much assay should be there, how much impurity should be there, what is the drug release. So for official product that limit is there, but for non-official product, for example, first time I am manufacturing a new product. So what will be the criteria? So it depends upon the. So for example, I am manufacturing a generic product, right? So what is generic product?
Expired one.
No.
Innovator.
It is not necessarily that innovator is expired. When we are making F2F formulation, first to file formulation, in that case, we will invalidate the innovator patent. Whatever he is claiming, innovator is claiming in the patent, something he will claim, right? So what we will do? Invalidate. Whatever he is saying, we will prove that whatever he is saying that is wrong. We can go with this way also. Or non-infringing. Non-infringing means, for example, innovator has patented like disintegrant to be used in the range of 5 to 10 mg. So if I am using 15 mg, then I am out of patent. So I am making non-infringing formulation. Okay. So generic products are normally high quality, low cost alternative for innovator product. So one of the best example, yesterday only one news came that I have seen. Risdiplam, one drug is there, that is a orphan drug. It comes under orphan drug category. RLD, innovator cost is 33 lakhs for 1 month therapy. Okay, 16.5 lakh for one bottle. 2 bottles are required for 1 month therapy, and that is for a chronic disease. So in India we can't expect, right? So there are only very few people that can afford that much cost of single medicine. Along with that, other medicines will also be there. So one of the company from Hyderabad only, so it is competitor to Hetero, so I will not tell the name. Okay, so they have already got the approval and they are launching in only 60,000 rupees per month. So cost of therapy is only 60,000 when compared to 30 lakhs of innovator, means 98% less. Means if you are giving 100 rupees for RLD product, for generic product you have to give only 2 rupees, right? So for generic product it is not like that, it is of low quality. Why? Because they have done the bio studies. Bio studies means pharmaceutical equivalence studies. They have given both RLD product, innovator product and generic product to the healthy volunteers, and they have shown that whatever plasma drug concentration they are getting in case of RLD, same plasma drug concentration we are getting in the test product. So our product is bioequivalent, like that.
Yes, thank you.
Thank you. So bioequivalence criteria is plus minus 20%, right? Okay. So whatever innovator is showing during the plasma level time profile curve, we should be plus minus 20% only. So plus minus 20% is not 80 to 120. Okay. Anybody can answer? What is the actual limit and how it comes? So it is 80 to 125. Okay, because it is T by R ratio. T by R ratio means if you are if your test product is 80 and reference is 100, then your result is 80%, right? But if your test product is 100 and reference is 80, then 100 divided by 80 comes 125, right? So that's why the actual range is 80 to 125%. So that comes by T by R ratio. It is not 80 to 120. Normally people will say that plus minus 20 means 80 to 120. It is not 80 to 120, it is 80 to 125%. Next question. Anybody else? So from my side, I will add something. Anybody is having any question?
Yeah, thank you, sir, for your presentation. As they have given introduction about you, all the achievements you have achieved, sir, for the benefit of the audience, can you say how your journey has been and how you were able to come to this position? As a young B.Pharm students, what can you do in our various students?
Right, so that's what I was going to say. Okay, thank you. So the basic thing is that in when whatever is your target, you have to first select what is your target. Either you want to go to industry, go to job after doing M.Pharm only, or you want to go to post graduation. So for industry, for research and development, if you want to go to research and development, whether it is formulation research or development or regulatory part or IPR part or analytical research and development, you should have post graduation. Minimum post graduation is required if you want to have some good position or if you want to start with the good company. So better to have the post graduation if you are targeting research and development and related departments. Okay, and if you want to go to manufacturing, then also if you are having a postgraduate degree, then it is better. But because of some reason, if you can't afford to do post graduation, that may be financial or anything, that may be anything. If you are not able to do, then B.Pharm is okay. But if you want to start with a good position, good company, and further you want to excel in the whatever you are doing in your job profile, so you should have the higher qualification. It is better because whenever comparison will be there, the basic thing will come that whether he is a B.Pharm or M.Pharm, whether he is MSc or B.Pharm, M.Pharm, MSc. So the comparison will come, and that is for initial thing. And after getting the job, you have to prove yourself. Okay. So whatever degree you are having, if you are M.Pharm, but if you are not having more knowledge when compared to MSc, if one person is from Master of Science degree, if he is he is having more knowledge, so many people will be there, who are MSc but they are having more knowledge than B.Pharm and M.Pharm. Many students will be there, B.Pharm students, that they may be having more knowledge when compared to M.Pharm. So it depends upon person, but you have to be focused. You have to be means this is the time. B.Pharm only is the time that you have to select what you want to go in the future. If you want to go into manufacturing, you can go with B.Pharm also. But better to have higher qualification, whatever is possible, and be dedicated. Whatever you are doing, you have to do it perfectly, as much it is possible. Thank you.
Good morning, sir.
Good morning.
Thank you for your speech. It was very valuable and informative. So I have a small question, sir. So we as pharmacy students, we have so many options in future, like who want to go to job. So, in hetero labs, like what are the current trending jobs so that we can easily get through them?
Okay, thank you. Nice question.
Also, one more question, sir, like.
First, I will answer this one, then we will go to next question. Okay. Definitely, I will address that question also, but I will first answer this question. So, as far as hetero is concerned, so it is similar to other organization like Dr. Reddy's, Arvindo, MSN, or all other companies. So, these in Hyderabad only, these are having manufacturing units where all manufacturing related activities are done. So for pharmacy student, that can be in terms of manufacturing plant, it can be in a manufacturing location, it can be production chemist, right, QC chemist, IPQA, in process quality assurance. So all those will come into picture if you are going for manufacturing, right? And others are related to R&D. Related to R&D means formulation development, where pharmaceutics is required, analytical research and development, where chemistry background is normally required, right? CPD, clinical pharmacology department, where these pharmacokinetic studies are done, their pharmacology is required. IPR is there, intellectual property right department, that will look into the patent and all those things. So normally, for example, I am from formulation department. So what I will do, I will make a strategy by looking into the patent, but ultimately I will send my strategy to IPR department for getting clearance, because those are more specialized person in the organization to look into the patent related matters. So I will formulate, I will give the means tentative formulation, tentative strategies I will give to them, and they will clear what is the strategy that you can go ahead. That may be, for example, if I am giving 4 options, they may give what is the preferential strategy, or they can say no, all 4 are infringing only, we can't go with these strategies. So we will get the clearance from IPR. For getting pharmacokinetic studies, we will submit sample to CPD, our pharmacological department team. So they will do the studies on human volunteers, and they will give the result that whatever formulation we have manufactured, whether it is bioequivalent to the reference product or not. If it is not bioequivalent, then we have to again go to our formulation, and again we have to manufacture new batch. What was the reason that we have to see, and to overcome that reason we will give the probable solutions. So okay, all these department works hand in hand to have a successful formulation in the market. So next you can ask.
Okay, sir, thank you. And sir, regarding internships, like how can hetero labs can help us, sir? Any procedures?
Hetero internship, you can directly apply in the website. HR address is there. Okay, and I will tell my mail ID also. So you can forward CV mentioning what is your requirement. If you are a B Form, you are looking for internship, or if you have completed B Form, M Form, means what is your present qualification. Based on that, I will forward to HR, or directly you can forward to HR also. So normally in hetero, what they are doing, they are taking as an intern for both B Form and M Form students. So B Form students, a month-end program is there. Okay, so 2 to 3 months training will be there, and along with this stipend, training will be there. You have to attend the classes and perform. Based on performance, they will make the grading, and based on grading, based on requirement, they will further send the candidates to the respective department. So MFOM also, they are directly coming as an intern. So for MFOM, there will be no classroom training. They will be directly joining to the department, working as an intern. And but in case of MFOM students, after completion of their internship, if the position is there, vacancy is there, then only interview will be conducted and you will be selected. Otherwise, after completion of internship, you have to move out. So there is no 100% guarantee for MFOM student. But basically, if your performance is good, normally 99% case you will be absorbed in the organization. But it is not 100% that if you are doing internship, you will get the job. It depends upon so many factors, right? So based on requirement, based on your performance, all those things will be look into it. Thank you.
Thank you, sir.
Any other question? Okay.
Thank you, Dr. Ankithanan Kharia, Assistant General Manager at FRME Hetero Labs, Hyderabad, for sharing your rich industry experience and research expertise, offering our students valuable perspectives on formulation development and evolving landscape of pharmaceutical R&D. Now, may I now take the pleasure to call our Dr. Arunava Malik to honor our guest, Dr. Ankithanan Kharia.
Sorry, I forgot to mention my mail ID. You can note down and you can connect with me anytime. Ankit.k, ANKIT.k @hetero.com. Okay. Number also you can write, but it will not be having WhatsApp. So you can message or you can call in the office time. 9100 129 449. It's 9100 129 449. Okay. Thank you. Thank you all.
Thank you, sir. Now let us take a short break. I request everyone to come back within 10 minutes.
Please stay back, please stay back, please stay back.
biotech startup housed at AIC Hyderabad. The company is focused on developing innovative vaccines and allied therapeutic solutions, working toward establishing proof of concept in next generation healthcare technologies. With a remarkable professional journey spanning over 3 decades, Dr. Raina brings with him extensive expertise across scientific, operational, and leadership roles in clinical, bioanalytical, and analytical research. He also serves as a scientific advisor to Asian Drugs Hyderabad, lending his deep insights to guide research strategy and development. Over the years, he has contributed significantly through his work at prestigious organizations such as Nectar Therapeutics, Sun Pharma Advanced Research Center, and Multichem Research Development, eventually evolving into a pivotal role in clinical research where he established several cutting-edge labs supporting both non-clinical and clinical studies. Dr. Raina is particularly recognized for his expertise in bioanalytical assay development for both small and large molecules and for pioneering high-throughput lab technologies. Notably, he introduced dry blood spot technology with an integrated Watson LIMS digital platform to streamline drug discovery research. A staunch advocate of ethical business practices and regulatory compliance, Dr. Raina is deeply committed to the principles of GCP and GLP. For over a decade, he has been actively involved with applied pharmaceutical analysis under the Boston Society, promoting scientific excellence through workshops and conferences. We are truly honored to have Dr. Vijay Kumar Raina with us today. May I now invite him to share his expertise and insights with us.
Shall we start? Yeah, sure. No, this is good. Good morning to all. It's an honor to be with the students always. I like to interact with students because you are the future generation of India. And most of you may join the industry, some of you may join academics and all. So it's better that learning, sharing of experience and learnings always should excel your growth, career growth. The difference between our time and your time is that technology is around you actually. Technology and innovation is growing so fast in the global scenario that it's very hard to catch up with the technology. By the time you learn something, the technology is again moving forward. So you must have seen in recent years also that the digital revolution has come throughout the world, and accessibility to the technology is almost happening in every village. And in our time it was not possible because there were no phones, there is no internet, so we have to go to the textbooks and then read, and then leave aside the reading. We don't have even access to the modern instrumentation at that time. But that's okay. So I just want to tell you that I'm going to touch upon the basic concepts of pharmaceutical research. What is drug discovery? I'll touch upon that topic also, and then what are the current technological interventions that are changing the pharmacy business across the globe. And of course we are part of the ecosystem. India is part of ecosystem because when you talk about the global technology, global innovation, so we are also part of that because ecosystem is such, we can't live without any other part of the world. Otherwise we'll be left behind. So what I mean to say is, I told you your time and my time. I just want you to go through this slide. These are wonder drugs. all the 4 drugs have won Nobel Prizes. And if you go back and see the dates on these drugs when they were discovered, they are discovered accidentally also, and there is no technology, there is no, tools whereby you can design the structure of a drug, or you can think that okay, this is going to happen, and then you don't have a predictive model in those days when these drugs are discovered. But if you see, these drugs are still relevant. Even after 120 years, 130 years, these drugs are in a way most selling drugs in the global pharma business. Morphine, if you know that this is given to cancer patients when they are in chronic pain, actually, chronic or acute pain. Aspirin, we all know that this is given for different conditions. Now most of the people are using it as a blood thinner. And insulin, India is fast catching up with disease like diabetes because of our lifestyle changes. Because of we are not the food that we are eating is not compatible with our body sometimes, and then there is less work that we are doing right now. That's why there is an apprehension that India may become the global capital of diabetes in next 5 to 10 years. So this insulin is still relevant, whether the source of insulin is from any other protein species. But insulin as a whole is very important drug for our society. It saves millions of people from getting into complications. Similarly, penicillin. Whatever drugs we have in the form of next generation antibiotics and all, their basic constitution is penicillin. So the question here is that if we have 4 drugs which are still relevant today and all the 4 have won Nobel Prizes, there was no technology at that time. It was all either non-existent, and there were no assay measurements. Only these have come into this world, these drugs have come into this world by chance actually, and by the dint of hard work of scientists who were thinking that if we use this, like insulin, if you see, the first insulin drug that was, protein was generated, it was given to a dog, pancreas and other things. It was tested on the dog actually. So what I mean to say is, advancement happens actually. So innovation doesn't stop actually. If there is some innovation happening today, it is going to happen tomorrow as well. So what we need to do as a scientist is that we have to catch up, read whatever is good for us, whatever is not good for us, we should adopt whatever is best out of that technology, we have to adopt if we really want to move forward. And even a college like yours, there are so many colleges in the city around, even there is a BITS, Hyderabad also, your college, CMR, there are so many multiple colleges here. So if all of you don't catch up with the latest technology and latest understanding of scientific knowledge, so then we are going to left behind with that. We are not going ahead, we are putting ourselves in a situation where we are not learning anything. I'll tell you what has changed from the technology. Even I would say in my time when I was studying or I was going into the lab, if we say procedures, all procedures were paper procedures. There are certain companies still who are using the paper procedures, but now the change has happened, it has gone to digital. With the advent of computer technology from 2000 onwards, last 20-25 years have seen the digital transformation. So if you have to take a training, say for example, on an SOP, suppose there is an SOP, how to, I would say simple, you may be using a weighing balance in the lab, and or any small instrument for that matter, or a pipette. So if you have a SOP today, there is a written procedure, SOP, why this SOP is there? There is a SOP scope is there, a procedure is there, why this SOP is there, who are the people who need to be trained on this SOP. So this is in the paper format. There are certain institutions where still paper is being used if they have not gone digital, because there is always cost involved with everything. If you embrace a technology, cost is involved. Whether your institute is really capable to handle that much of cost, and with the advent of technology, are you going to upgrade every day? It's not possible actually. Frankly speaking, it's not possible. So there may be some institutions where even paper procedures are still used, but there is an alternative of digital procedures also. There are so many programs which can be uploaded onto the digital platform, and then what you do, you can go home, you can open your computer, you can take a digital training, and then take an assessment. There are certain platforms like that. That's why I am telling you that this paper to digital transformation has already happened. Whether your college is aware of this or you are using, or is it a requirement for your college? We don't know actually, because it also depends on if you have a requirement, then you can implement that procedure. So that's what it is. In the procedures, in the paper, from paper we have gone to digital. But when you get, see, now you see the paper, I'm trying to give a concept that you must understand. If we are having a paper in hand, unless it is signed by somebody who is authorized, suppose it's not signed by the principal, somebody, a person like me will come to you, oh, this is the new rule that has come into force in your college. Are you going to accept it unless it is signed by the principal of the college or any other authority of the college? You are not. I can even type it and show it to you. So that's not the way we should accept the things. There has to be a signature of some authority on the circular notice, then only it's valid. Similarly, when you have digital platform, there is an electronic signature on that. Unless there is an electronic signature, it's considered a draft only. It's not implementable. That's what I mean by e-signature. Now, when I'm talking about digital, where do you store this? We cannot store this on a computer which can be hacked by everybody. There may be some confidential information in this, so data security is very important. I'll come to that in later part of my presentation also, why data security is very important. I am using certain terminologies which are very, very important for D Pharmacy and PharmD or MPharm students to make a note of it, because these will help you to face an interview, write an SOP. If you are taking an admission into higher classes and all, you should note these terminologies, what is happening, because the theme of this whole program is innovation and technology. So you should remember these things, data security. Then storage, as I told you that we cannot store this simple digital copy on a simple computer. It has to be secure. If a person who is running the IT, he need to have a login and password to access. Every person or every student cannot go to that computer and have an access. So it's not done like that. There has to be a proper system even to run a digital platform. That's what I mean by data security, storage, retrieval. Suppose you do some work today, and after 10 years, suppose you are filing a patent or you are doing something which is very unique, and your college has the ownership of that particular research. Small research also is sometimes a big, causes a big impact on the public life. So if you want to see that what had happened 10 years back, where do you store your digital platform or a paper? So security, data security, retrieval, archival, traceability, these are all important aspects, whether you do manual or digital, those are processes. But I just wanted to tell you what is digital and what is manual. That has happened in procedures. That transformation, technological intervention has happened in procedures. Now, what has happened to instruments? If you have seen a HPLC, in our times, we used to prepare the sample. Suppose you have to do a formulation. Suppose you want to do an analysis of water. Say, for example, let me tell you whether this contains some impurities. So I go to HPLC, I prepare a sample and then inject it with my hand. And there is a injector which is just like a small syringe. I just see with the eyes that okay, I have to inject 1 ml or 0.5 ml or 0.2 ml. So what is the accuracy level is not that precise. It's not that accurate and that precise. Suppose I may inject even 0.2 ml also, I can inject 0.19 ml also. So that's the way we used to do, like inject one sample, see the result, then inject again. So we have to sit in the lab for 8-9 hours to do the manual injection only. There is no other work that we are doing. But now the system has changed. Auto injector has come, auto sampler, which is unattended operation. Suppose you prepare 50 samples and put it in the auto sampler, give a command, HPLC will do its job. And till the time your analysis is complete, suppose runtime is 20 minutes. Are you able to understand what I am saying, or just I am saying something which is not of your interest? Are you sure?
Yes.
Okay. So if I am putting a sample, so I can go and make a report actually. See, time that is spent on the manual injection, now I have that time to write a report. I have time to gossip with my friends. I have time to go to the library. I have time to say hello to somebody. So that's also important. See, social interaction is also important. When you are working in this college or you are studying, everything is important in life. Don't think that only study, doing research is important. Every aspect of life is very, very important. And that's how a happy and healthy living comes into being. So you should always enjoy when you are coming to either college, go to market, go to your home, always be happy. And that happiness comes only when you live a holistic living. So I just wanted to give you that perspective, what a manual injection and a auto sampler injection is. Is that a digital transformation or not? Over a period of 20 years? So I am giving you just small examples. Similarly, when I was dealing with NMR, I was doing NMR analysis, so it was only 60 megahertz. We used to prepare a sample in D2O, deuterated this water, and then we used to put the sample in the tube and then shake it, put the vial in the NMR probe into that machine, and then see how it is giving the carbon, hydrogen and other peaks and all, and then we used to interpret. Is this peak for OH, is this peak for something like that, carbon-carbon bonding, CH bonding, CH2 bonding, something like that. But now it has transformed into the digitization and innovation has brought it to 500 megahertz, which is almost 10 times more accurate and precise, and with 3D technology. So you can see a compound, virtual compound, you can see in the computer itself how a structure is looking like. That's what digital transformation has done to the technology, has done to this pharmaceutical research. If you see measurements also have taken a big leap actually. Because when instrumentation, the sensitivity of the instrument goes to a higher level, so you can measure a small thing also with that kind of equipment. So when I was working, it was ppm, milligram. ppm is nothing but milligram. It went to micrograms. So we used to have, suppose I do an assay of metformin. It was 0.1 microgram, which is 100 ppm, 100 microgram. So 0.1 milligram scale we were measuring, whether it's a formulation or it's an analytical technique or whatever. Do you know, now if I go to LCMS, if I am doing the same sort of analysis on LCMS, we can go up to nanogram, picogram level. If you have heard a drug, during the COVID time also, everybody has used these inhalers and all. So some of the old people, they have used inhalers, budesonide, Flucot and all, which are available in the market. Do you know when the trials are happening for those kind of drugs? So you give 1 puff or 2 puff. 1 puff is only say 10 microgram. How do you estimate that sensitivity? It is in picogram level. Picogram is even the last one. microgram is 10 raised power 3 and nanogram is 10 raised power 6. This is 10 raised power 9. 10 raised power 3, 10 raised power 3 is milligram, microgram is 10 6, 10 9 and 10 12. Minus 12. I am telling you one, if you say one part, out of that 1 billionth part you are measuring with high-end instrument like LCMS, which is very high. Now a lot of LCMS machines, there was a series 2000, 3000, 4000, 5000, 6000. It's just like those antibiotics which have penicillin as first generation, then next generation streptomycin, third generation, something else, and like that it goes. So even instrument series are like that. If you make one upscale in any instrument, it is next generation. So similarly, you can imagine now, if you have to measure one puff which is given in the nasal route, how do you measure that? Those kind of instruments are also available. They may not be required for you in this institute. It's not required because they are commercial instruments. So that's the level of detection that has actually transformed the technology from that time to this time. Similarly, regulations. This is very important. I want to spend a little bit of time here. See, do you know how are rules framed in a society? Rules are always framed by the government, right? Suppose there are more accidents happening, so then traffic police will come and they will tell, no, we have to build a footpath, and then we'll not allow people to actually drive more than 60 kilometers per hour speed in a city limit or something like that. Rules are formed when some problems happen. Rules are not formed just like that. Similarly, regulations, because drug manufacturing is a big thing. Once there is a tablet of say 10 milligram, 5 milligram, 500 milligram, 1000 milligram, this may cause a death also to a human being. Because if impurities are there, if we don't assess the impurities, I just now told you there are instruments and which are required to measure even the most sensitive impurity in the drug. So if that impurity is not measured properly, you tell me, if we are scientists, if we don't measure properly, if we are not paying the attention, if we are not writing what we are doing, so what we are giving to patients is a substandard drug. So patient may die also. I may tell you, I have seen many people even getting allergies by taking even pantoprazole, which is very safe, proton pump inhibitor. You may know all these things. So some of the people are getting even reactions with a drug like aceclofenac also. Why it is so? Some of the people, there are some certain drugs which are manufactured by company A. They are giving reactions. They increase the heartbeat. I have seen it in my experience. If we take drug A from say, I will not name the company, but I know which companies are good companies. I know because I have that much of experience. I never use. When there is some prescription in my family, I take the prescription from doctor, but I decide which drug I have to give to my any family member. I don't take just like that what doctor gives. I do an intervention and then, because we don't know whether quality system is right and whether drug is manufactured by a company which has standard, which has ethical practice, which is following GMP practice or GMP practice. So those are important aspects, especially in India because our regulations are very weak. If regulations are written somewhere, they are not implemented. Our officers in the drug department are not so competent to see voluntarily what is happening in the market. So our system is little different than what it happens in US or UK or Canada. So what I mean to say is, so who is going to give the guarantee that if I am taking one tablet, whether this tablet is going to create positive effect or negative effect? Nobody is responsible for that. Then I have to prove in the court of law that I got an allergy because of this drug. Then pharma company will come and no, I have standard practice, it's not my fault. So ultimately patient will suffer. Patient is going to suffer, and if somebody dies in the family, we know what is the pain involved. What's the impact on the family? So there were some instances, because in those times when the drug manufacturing happened in US or countries like Europe or UK, there are no regulations at that time. People have done some misdoings also. Say there is an example that they had to use one chemical, they use other chemical and children died. 30-40 children died in US, and then regulators got into action. They told that why they have used this. They said we don't know. The company said we don't know. This was ethylene glycol PEG. They added PEG. They had to add something else. They added PEG, and then children died. Similarly, there is one more drug which caused genetic impact on the children. When children are born, they are either dumb. See, these kind of things have happened in US. At that time, India is not in the picture at all. We are ruled by British at that time. So, and we are not that much into allopathic. We have traditional methods at that time. So, if it can happen in US, it can happen in India also. So, that's how regulations have evolved over time. This started with in 1936, and now US, UK, Europe, Canada, Brazil, they have a robust regulatory system who are inspecting the companies, giving warning letters, even closing the companies if they find any discrepancy. If any Indian company is manufacturing the drug for US market or UK market, if I am a company owner, I want to sell a product in US, I have to do a clinical study. That's very, very important. I can't sell just like that. I can't make and sell. I have to do a clinical study, which is known as bioequivalence or bioavailability study. If there is a classification of drugs also, class A, class 1, 2, 3, 4, and if clinical study is not required, then at least prove through dissolution profile that okay, these are equivalent drugs. Those kind of stringent measures are existing there in foreign countries so that we can sell our drug into that country. That's one thing. Another thing is that these rules have come into picture because of mistakes, because of learnings. Always, I would suggest to you also, if you are making any error, don't be afraid. You should report that error to your supervisor. That's the best thing that you will do. Because if you hide certain things, you may be doing a good research, accident research, which may result into a good thing. So do not hide. If you are a good scientist, if you really want to become a good scientist, don't hide anything. If you make a mistake, you admit, I made a mistake. That's the best thing for sake of science, actually. That you should inculcate that habit that okay, if mistake happens, don't hide anything. So that gives you some confidence. Okay, I made a mistake, I learned from this mistake, next time I am not going to do. But if you hide, then it becomes a habit. So then we can't become a good scientist, we can't do justice to the science. That you should always remember. So in this slide, what I told you, how the transformation has taken place from old to new system. And this is not the end. Actually, this is not end. Tomorrow, in 10 years time, there may be something else coming in the market. So we have to be ready for that. What is the role of pharmaceutical industry in the society? Pharmaceutical, the role is that the first and the foremost thing is to develop the safe and efficacious medicine for the patients. That is an ethical requirement. That's an ethical requirement for any company who is making drugs. Do you agree to this statement or not? See, ethics is what? Ethics is if rules and regulations are not there. There may be a policeman standing at the red light signal and people stop. The beauty of that is that if policeman is not there, we should stop actually. For the respect of people who are walking on the road. To make sure that we don't hit somebody who is walking on the road in a certain condition. So that is what ethics means. Ethics doesn't mean rules and you have to follow rules and regulations. Ethics means what are you supposed to do as a human being, as a scientist, to help the society. So why we are doing this is for efficacious and medicine. See, when the drug is developed, it takes almost 15 years. I am coming to that slide also. 15 years and 2 to 3 billion US dollars, which is 15,000 to 18,000 crores of rupees. When you develop one single drug, you start with 20,000 molecules and end up with one molecule. From 20,000 to get one molecule, it costs around $20,000, 20,000 crore rupees. But a patient trial happens on 5,000, 10,000 people. We are 7 billion people in the world. So can you imagine 10,000 people are representation of 7 billion people? No. So there are certain indications when you develop a drug, they may be good for Caucasian population or Asian population. They may not be good for a white population or this African population. So there are certain things which happen post-marketing also. I am going to come to that slide also, what is the latest going on in that scenario. So it is always when a drug is given approval, approval is given based on the 5,000 patient data or 10,000 patient data. Because regulatory people also don't know that this drug is going to make to the market and what impact it will have. Therefore, post-marketing surveillance is very much required, which very seldom happens in India, but in US, UK, in European countries, it happens with complete dedication and there are procedures for that. So that they come to know which of the drugs, say for example, this painkillers, nitrosamine impurities, 5-militazone is not sold in US because it has a serious cardiovascular effect. So how do we know that painkillers should be banned? Because post-marketing surveillance tells us, because patients have not, they are not compliant to these drugs, most of the patients. So people are withdrawing those drugs. Therefore, that's also one reason why research is happening. So we do repurposing of drugs. You may have heard that word, repurposing of drugs sometimes. We are changing the structure. Suppose diclofenac, now escitalopram came. There is some citalopram, escitalopram, because S is another form of stereochemistry form of citalopram. So those kind of developments happen. That's what these things are done only because of surveillance data. This doesn't happen just like that. There is a differentiated global disease burden. That's why pharmaceutical research is very important. Do you think from the first day, if we do any research, and if we have come to a certain level, regulatory people will never say you don't launch the product because they know risks are there in that development and all. But see, we can't stop the research also. That's why you may ask a question why regulatory people are giving approval for a drug which is tested only on 10,000 patients and they want us to use this medicine for 1 billion people or 2 billion people. It's not the case like that. They give you approval, otherwise research will stop. There will be no progress. They tell you, you do whatever you want to do, but tell us how it has behaved in the public after you have done the research. That's the essence of research actually. You can't stop technology, you can't stop any research because then world will not do anything. There will be no economy running around the research R&D. If companies are spending 10%, 15% amount of their profit on R&D, they have to do something. They have to bring out some technology out and they have to bring some drugs out. What is diverse global population need? See, there's a simple answer to this. The disease burden in US and UK is different than what it is in India. India, we have more infections, cancer is catching up, diabetes is catching up. So different disease burdens, when there is a different disease burden, so requirements of drugs are different. Therefore, our pharmaceutical research is concerned with some other form of research. It is drug research only, but more into generics, more into OTC products, more into those products which can be sold at an affordable cost to the public. So the focus is different because of diversified population. Unmet need, I told you, lifestyle diseases. I am telling you why pharmaceutical research is important and what are the fields we need the pharmaceutical research to happen. Multi-drug resistant infections you have heard. So this is because of erratic use of antibiotics. In our society, the thing happens like that. If we get a throat infection, we go to medical shop, not to doctor, to tell him give me some erythromycin or give me some other antibiotic. I want to eat 5-day course and all. So we are self-doctors actually, without doing pharmacy, without studying MBBS or most of us are self-doctors. So that has caused a lot of this in the society, like multi-drug resistance. And do you know why? Even quality of medicines. If people are not making quality medicines, that also creates a lot of problem. It can cause kidney failure. It can cause cardiovascular failure. People can die. That I told you already. Why quality is required in drug? Not only quality is required from the day API lands in some company to the product is made. Then regulations, it's surveillance. That whole ambit is quality. Quality is not just I am a scientist, I am doing my work properly. Everybody has to work properly, including the regulatory agencies. So affordable therapies, India is mostly into this. Emerging diseases, we don't know what is going to come tomorrow. See, there are some diseases like multiple sclerosis and most of the CNS diseases. We don't have any cure for them because brain is such an area, it's very grey area. So people have exploited this area in such a way they are selling a lot of drugs. There are almost 100 drugs which are in the CNS area, but we don't know the real effect of those drugs, whether they are doing some benefit to the patient or not. We don't know because we can't penetrate into the brain. The way if you are doing any study in animal or a patient, you can take a plasma sample and see what is the level of metabolites formed in this sample, and you can also see what is the level of concentration. But you can't go into the brain. You can't get the CSF, cerebrospinal fluid, to do the testing or see the online measurements of certain things. So it's not possible. CNS area is a grey area, so we don't have total control over the medication in CNS area. Those are emerging diseases. That's why, see, these are the main focus areas why pharmaceutical research is required. Now, what are the stages? Actually, discovery is in vitro testing. All of you must have heard this. Different phases of drug development. I told you 15 years of time and 20 to 24,000 crores of rupees are spent. And you need to have a skill set to do this research. Skill set means I cannot do, you cannot do, other person cannot do, unless in the discovery stage, in vitro testing, a chemist and a biologist are required. Then animal testing, toxicologist, veterinarian, GLP expert, all those people are required. Data interpretation, analyst, analytical development, similarly human clinical testing, GCP, clinical trial, doctors, investigators, principal investigators, all of these are required to do the clinical testing. Manufacturing, big plants are required to manufacture bulk drug, because if we start from here, this is milligram scale. If I say discovery, this is milligram scale to GMP scale is, it is in tons, because you are manufacturing for 1 million people in India. So GMP, it has to be of GMP standard. Even container, labeling, packing, this formulation, excipients, everything has to be of top quality. Then you can make a quality drug. And then marketing and surveillance, what I was telling you, this is the most important feature, because you cannot actually extrapolate 10,000 patient data on 1 billion or 2 billion people data, because we have diabetic patients, say 3 million or 4 million. So you cannot extrapolate that on diabetes patient. See, now I am telling you what is trending now. Trending is real world evidence studies. This is based on post-marketing surveillance data. So you take a data, you assign certain doctors, Suppose there is a small drug, 5 mg amlodipine or 10 mg amlodipine. You go to the doctor and do a study on the vast population, 50,000, 20,000 patients in a particular country, say for example in India. Then you monitor those patients for a period of say 5 years, 6 years. Then you have the real evidence, that is called real evidence of what this drug is doing post-market surveillance. Then we are making a particular recommendations to the manufacturing or regulations. We are making certain recommendations that this drug can be given only in the western part of India or in the southern part of India. Northern part of India, it has some issues, so it is not absorbing properly, it is causing cramp. So that is how this is also, this you should remember, real world evidence studies are catching up very fast because this is very easy to monitor. Already drugs are in the market, so this is no focus actually. This is a focus. So quality systems, what is required to do the discovery research or pharmaceutical research? Trained manpower. I told you skilled manpower. Unless we are skilled, we can't do anything. Qualified instrument. Qualified means if I am using a weighing balance, it has to be qualified if I am weighing 1 mg weight in the balance. So whether I am weighing 1 mg or 0.99 or 0.98, that balance standardization has to be traceable to the primary standard which sits in Geneva. So when you do the weight calibration, you have to do the weight calibration of your weights with the Geneva standard. There are some laboratories in India also, National Physical Laboratories 1, NPL. This is in Ahmedabad. So when I am doing the lab activity, so we are taking those weights to NPL, give them the weight box, they will weigh it on the primary standard and then give us whether your weight is 1.001 or it is 0.998. So then you calibrate your dilutions accordingly. So to that level, it has to be just like that. So that is what qualified instrument. I am taking the example of only weighing machine. Suppose you do HPLC injector, how much, if I am injecting 10 microliter, whether it is taking 10 microliter or 9, that is the qualification that I need to do for an instrument. It's very simple, it's not very complicated, but procedure, we have to learn those procedures, execute those procedures, because if it takes only 9 microliter, that 9 microliter gives me suppose 10 picogram peak. That means 9 is equivalent to 10. It's not equal to 10 actually. It's equal to 9 is equal to 9 only. So those modifications we need to do when we are doing qualified instruments. So we are close to accuracy and we are close to precision. SOPs is also very important. I told you example of SOPs in the beginning only. Quality assurance. Quality assurance should be independent team. Suppose I would say that if you are pharmacy, there are so many sections in the pharmacy school. In your pharmacy school. So quality assurance is just like this. If you have a team, independent team, these three gentlemen are sitting here. They come to your lab and they see whether cleanliness has happened. They have nothing to do with your department and all. We appoint them, our principal appoints them to do a checking. Whether washrooms are clean, whether food you are getting is clean and all that. That's auditing actually, independent auditing. That's what quality assurance means. They are independent function. Their job is only to check whether what you have written in the procedures is followed or not. Then data integrity is very important. I told you in the beginning only, I am mentioning certain words just for your attention so that you know what data integrity is a big issue. Data integrity is that whatever you see, whatever you observe, you write that. If you see 7.2 milligram you have weighed, you want to weigh 9 milligrams. Say for example, by mistake you weighed 10.01, that's okay. But you record that. That's known as data integrity. If you are weighing 10 milligram, you can't write 9. If you write 9, that is violation actually. That's a violation. That is not ethical conduct. So you should understand what data integrity means. What you do, you write exactly the same. Nobody is going to harm you. At the most, you can say, sorry, I made a mistake, but this is what I have weighed. You understood or not? I am trying to give you in the very simple form so that you understand the conversation well. There are interdependencies. Only one person cannot do everything. If you see this, NC design, who are all involved. So we often say in big institutions that interdisciplinary. So this is everybody's responsibility to do the best. Like in a family, suppose the elder of the family, he earns money, and then our mothers cook food, and then somebody washes clothes, somebody. So everything happens. Can one person do all the things? It's not possible. If you take a small family also, we have to share the responsibilities. If you really want to make a happy living, and also not to burden one person with lot of responsibilities. That's also not fair. So similarly here also the same thing happens. If you have NC design, chemistry, analytical chemistry, then biology, bioanalytics, product development, PK talks, clinical regulatory, these are all essentials of pharmaceutical research. And I have also listed what activities are being done. Say for example, process characterization, purity content, impurity profiling is very, very important. I am telling you, if you take a drug, suppose you keep it in your home for 6 months, 7 months, the tablet looks white only because we are just taking out the tablet and taking a glass of water and taking it. But what is the guarantee? Intrinsic reactions have not happened. If these tablets have some kind of solvents left behind, which are not properly dried during the product development phase, then do you know what will happen? They will cause mutagenicity, carcinogenicity. We don't know actually. After 10 years, 15 years, people may develop cancer also, because there is a gene mutation happening with some of the chemicals like benzene, toluene and all. So if we are not taking care, you see how important is this? If we are not taking care of the manufacturing process in the way it is written on the procedure, and we are not doing it as per the guidance, we are, instead of doing justice to patients, we are killing them actually. So how much important is this? Now the question is impurity profiling also. There are, because of instrumentation, advancement of instrumentation, now there are so many instruments where you have to actually show to the ppm level or nanogram level that what is the content of impurity that your product is having. You have to report that. You can sell the product, not an issue, but you have to list that impurity in the CTD. CTD is a common technical document which is submitted in the for the approval. So we have to list everything. It takes almost 4 months, 5 months for regulatory people to read the report actually, and make a marking, put yellow stickers and all. Then they call the manufacturers. They tell them why this impurity is this much, what is your mitigation plan to reduce this impurity and all. See, lot of discussions happen there. See, if we see a policeman in India, what do? We fly. We don't face that policeman. In US it's not like that. When you go to any inspector, regulatory, they take you, they are helping hand actually. That's why in US industry nobody is afraid of inspector. They will show you the way how to correct your things. Even if you made mistakes, they will show you the ways how to mitigate the risks and all. Here we are hiding because that's in culture actually. We are hiding. If we see something wrong, we are hiding. That's why I sincerely urge you, don't hide anything. Because some of you may go to US, you may go to Germany, Europe to do jobs at later stage of life. You will learn. Those kind of things are very prevalent there. So why don't you train your brain in such a way so that, train your mind in such a way so that you get into that habit of reporting proper things with data integrity and all, so that your life becomes easy when you go to the industry. My point here is only that we are dependent on each other. If one of us makes mistake, everything is screwed up. And I told you what is the cost implication and what are the quality implications on this. So please try to help each other. We are doing a research which is interdependent on each of us. So we all have to do our best. Even if we don't do the best, report what you have done actually. That's the essence of this slide. You got it, or I am boring you? Okay. Now, quality control requirement. These are all important slides. Actually, I made this with a thought process. Whenever I go anywhere to give a lecture, it is always new lecture. It's not old slide or something. Because I have to also prepare. And if you believe me, honestly, I have worked almost 12 to 13 years cumulative to make this slide here. Because I cannot just come and go, and then I have to prepare for something so that you get to know something. And in the process, I am also learning. Because when the new technology is happening, so I did not know certain things. Through this slide deck only I came to know, I read, and then I put this in the slide. So this, when we talk about the quality control is what? The quality of data that you are producing. See, the first, whichever is in the green, we should always target that, which is precise and accurate. Rest of the points are, see, if you see the last one, fourth one, this is inaccurate, because accuracy means in the middle. We have to be closer to the true value. That is accuracy definition. Precise is the reproducibility. There are only two things required in quality control system. One is reproducibility, one is accuracy. Reproducibility is nothing but precision. Accuracy is what is the closest value to the true value. So those are the three things, two things which are essence of any quality control system. When you are doing assay development, when you are doing any experiment, you should repeat it once or twice so that you get a confidence. If any senior comes and then he wants to repeat your experiment, you should write your experiment in such a way so that it is automatically reproducible. So that means procedure that you are writing should be written very carefully and very simple language. If you have to weigh 20 milligram, you weigh 20 milligram, I weigh 19.98, put it in the 100 ml of water, use this beaker or you use a flask. That is very important, because beaker accuracy is little different than the volumetric flask. You understood? Whether you weighed by pipette or burette, especially those things are very important, because accuracy level is different. So this is what is quality. So precision and accuracy is also very important. Yeah, you can just do it from there. I want to show you this slide, what is the transformation happening in the world. The first slide is 2010 slide. So I am showing the top 10 selling drugs in the first box. Second box is top selling drugs in 2020. Third slide is post-COVID. What is the scene? You don't need to read this, don't need to remember. This is all top 10 drugs. See, at one point of time in 2005-2006, atorvastatin, which is given, which is HMG co-reductase inhibitor, the largest selling drug, that was the largest selling drug, because people are afraid and doctors are taking full advantage of insecurity of people. They are prescribing like anything, 10 mg, 20 mg every day, otherwise you will die of heart attack. Nobody is going to die of heart attack because of atorvastatin. Do you know why? Because atorvastatin lipids are also required for brain development. There is a probability that if patients are taking more atorvastatin, their later part of life may lead into dementia also. That's also one theory. We don't know. There is no authentic research on that, done on that. But lipids are also important. The only thing is, because of insecurity, certain people are trying to sell the products just like that. There is a lot of controversy around atorvastatin also in US. Like doctors have unnecessarily prescribed those drugs. However, let us focus on the agenda. These are the 10 drugs. First, I want to tell you what, there are two types of drugs. One is small molecule, one is large molecule. Small molecules are common drugs like aspirin. Insulin is a large molecule because the molecular weight is more. When we have molecular weight more than say 2K, 1.5K, Dalton, 1500 Dalton. This goes to the large molecule category, and mostly large molecules are protein molecules. They are complex, they have a complex nature because this is made of amino acids. This is made of simple chemical, benzene ring, aliphatic ring, some groups and all. That's the difference between small molecules and large molecules. The slide that I want to touch upon here is, there is a shift happening from small molecules to large biotech molecules. That's the shift. And post-COVID, we have top selling vaccines also in the world. That's the shift post-COVID that has happened. What is trending here? This is also because we are discussing technology and innovation. Biosimilars are trending. So your focus, you should also try to go to the websites of top companies like Genentech is there, Gilead is there, Merck is there. Go to their website, don't do anything. You have a lot of information on these websites, how the research is shaping up for the next generation. You should go to these websites and see what these companies are doing, how much they are spending on R&D, what are their capabilities. See, this is simple, you all have mobile phones and all. Go to top company websites, Pfizer is there, Gilead is there, Eli Lilly is there. What are they doing in the research? Go to their research page, what are their publications, who are the scientists? See, that's very important. When you have time, go to that website and see what is happening. Go to Nature publications, go to Journal of Pharmacy and see, not the Indian journal. Indian journal, you won't get anything. Go to the foreign journal and see how much. And I can tell you one thing, even in foreign journals, there are a lot of Indians who are doing best work. You will see one or two names, Indian names in those journals also, even if it is Nature, Communication, anything. So you just try to spend your time, leisure time on these things also. Then it will add a lot of value to your education. And understand, there is a lot of information. That's the advantage of digital technology. There is a lot of information which is free of cost available to us. So make use of it. So what I mean to say here is, there is a global shift from small molecules to large molecules, and now biosimilars are catching up, and even vaccines are catching up. Because see, COVID is going to stay. This is which many people believe that this is a man-made virus actually. We don't know who made it. People at the top level may know, but we don't know as a common citizens. Otherwise, how can this is a combination of that HIV spike with pneumococcal, this combination of virus and all. This is very dangerous actually. See, millions of people have died across the globe, and there is no cure for it. See the situation in 2021, how many people have lost their loved ones and all, just because something has happened somewhere. So we lost a lot of people. So I want to touch upon this topic also, lessons from COVID is that because there is unprecedented demand of vaccines. There is no cure. People are giving some of the drugs, even Dolo 650. There is no cure to virus. All of us know. We should know. We are pharmacy students or PharmD students. There is no cure for virus. It is only autoimmune system that is going to take the advantage of this and then develop an immune system. Some of the scientists even suggested that let us have the herd immunity. It may save a lot of people, because drugs are not, that remdesivir and all, they are not of any use actually. That's only insecurity. People have sold that injection for 50,000-60,000 rupees, which is costing just 1,000 rupees. So that's the kind of insecurity society we see in the society happening. Shutting down of clinical trials. I'll give my example. I was working in a US company, so we were working on cytokines, interleukins and other things. So I had samples from 10,000 patients in my lab. 3,000 patients had died actually because of COVID. So we didn't have, we had only 7,000 samples or 8,000 samples which were from healthy volunteers. Now, if our p-value can be determined only by 10,000 plus patients, how can I do a justice to the 7,000 data points? So we shut down our clinical trials. That's what has happened in COVID. because it is linked to cytokine syndrome, COVID, and we are also doing the same kind of research. So unfortunately, we have to shut down 2 clinical trials. Shortage of PPEs. So now India is the largest manufacturer of PPE, but at that time we don't have PPE. We don't know even why PPE is required. And this COVID has also changed. When you see the last slide, there is introduction of vaccines into the highest selling drugs category by Moderna and Pfizer and all. And even our own Bharat Biotech, which is SCT based vaccine company. So they are the largest vaccine sellers in the globe. Actually, they are supplying to WHO and all. So we have one, Dr. Krishna, who is owner of Bharat Biotech. They did a lot of work, putting up a lot of efforts, actually working continuously for 72 hours. They are scientists and all to develop this vaccine to save Indians from the disease. My own thinking was, because of insecurity, people with COVID infection died. So could it be a situation that because we are all actually scared, if we put those patients in a common ground, have community kitchens and all, I think things would have been totally different. Because there is no cure. What to do with these patients? If they need a community support, they want to talk, they want to be reassured that you will not die. But instead what we do, we dump them into the hospital and they died without seeing even their children. This is my own thinking. We could have done differently, but we didn't do. We could have saved a lot of people by doing this kind of community treatment, I would say. Because there is no cure. If there is cure, we can take a person to the hospital. But there is no cure in the hospital. Hospital is charging you 1 lakh rupees per day. How can a poor person go to a hospital in those situations? So, if we put them in the Secunderabad ground and call all the people, volunteers, to serve them, at least they have somebody to look after, and there will be a lot of people who will save from this pandemic. That's my own personal thinking. So, digital technology, how has digital technology, I gave you a little bit of flavor, how technology changes, what are the changes. Now I'll come to simple, this is, now boom has come. See, if you all go to the chat GPT and then tell him that you write a letter for me, for the principal to give me a grant or give me a leave, you will get 15 letters from chat GPT. You must have all used that. Actually, AI is creating a boom. Actually, AI this was 1956 when the AI first concept was conceived by one scientist who was developing the computer. Later on, it became machine learning, deep learning, and now GI. GI is the latest technology. See, what has happened with AI? You tell me. When the advent of computers happened in 2000, from 2000 to 2020. There is lot of data which is our own data, which is stored on the servers. So what these wise people did, who are business leaders in the global scenario, what they did, they used our technology, processed it, created an algorithm so that it gives an answer to our own questions. What we have fed on the system, on the cloud, they are using that only for us to earn money. That is what AI is. Then why they will call it artificial? Why it is not called intelligence? You tell me. They have very carefully chosen this word. This is artificial intelligence. Human intervention is always required. And this, two people have won Nobel Prize on artificial intelligence in 2024. But there is a caution that red line is there, ethical practice. Because when you have a lot of data, who is the owner of the data? That's the discussion going on in Europe, America. Lot of lawyers are discussing, lot of scientists are discussing. Suppose you create a protein sequence with help of GI. Who is the owner of that protein sequence? It is already there. Somebody must have worked 20 years before, but ownership, patent right, that's the big challenge now actually. So this is going to come. And most of the technology which comes in the world, in the globe, it is misused. It is used for goodness to a very small level, but most of it is misused. We have to see this how shapes up. And fortunately, India is also like one country which has adopted this AI very early on. When this new government came in picture, they actually grabbed the opportunity. They did not wait. Like we waited for many things. When ATM came, India waited for 5-6 years to see how this ATM thing is going on. People are still in the queue. But with this government, they have taken the initiative to catch up with the foreign countries because we are part of the ecosystem. If we are left behind, we can't generate innovation in our country. So that's one good thing has happened. Actually, I heard many people speaking in the government about blockchain way back 10-15 years back when it was catching up in US and all. So you have to catch up with the technology and take the best out of it. That's what I want to say. See, AI in pharma is, these are all small definitions. This is acceleration of research, helping scientists to generate hypotheses, design experiments, collect and interpret large data sets. That's what I was trying to tell in the first slide, that it's our own data actually. See, how many papers are being published globally? Thousands, millions maybe. See, this, what AI is doing is, this is capturing the information of that particular data. We are publishing papers. Even India is the third largest publication center for scientific journals. A lot of papers are being published from India also. See, all this information is going into the cloud, and there is an algorithm, there is a data processor which processes the data. That's how it is helping scientists to design a better design, an accurate design, where people cannot make a mistake. That's what. The second one is past decade breakthroughs, self-supervising learning. There is also, I am coming to that slide also, training modules are being developed for people how to do. Suppose you are designing now one experiment on chemical synthesis or biology. Now there is one more tool. So they are going to tell you how you are going to do your experiment. If you sit in home also and then take a training for 2 hours, and then tomorrow you go to the lab and you can remotely control your lab. There are certain human interventions required. Suppose you have to put something, some chemical in the petri dish and all. Those things you have to do. But that part also AI is going to take care. They are going to guide you properly. So then the question is, what degree we need to become a scientist? Do we need a law degree? Do we need a scientific degree? We don't know. At this point, nobody can tell us. So probably a art student can also be a scientist if we can use this technology. So that also you have to think. How future is going to become. Now we have voice modulation. If you have seen recently, if you call somebody, if the phone is busy, so they say voicemail. What is that? You tell me. Big companies are giving money to your this subscriber, this Airtel and other people, collect the voices of the people. Do you know that gang is doing their work? We are saying hello, when are you going to call me, and your voice is getting modulated. It may be used later on, 20 years down the line. See, those things are happening. This technology is very powerful tool, and then when somebody spends money into that, they want to, if they spend 10 rupees, they want to get 1,000 rupees out of that. You must remember that. See, but we are part of ecosystem. We can't just ignore that. This is what I was telling you, Chat GPT. This is a product of generative pre-trained transformer model. Chat GPT. GPT is this one. Chat is one more design which creates small molecule drugs and other. If you simply say, I want a protein sequence of insulin or insulin-like, you will get hundreds and thousands. Now it's up to you. If you are a biochemist, you can imagine if I change this, if I change that, what's going to be the impact? What's going to be the impact on the gene mutation and all? Those things are very impressive. But the question here is, who is going to own that research? Who is going to own that innovative idea? That's the debate that's going on in the at the global level. So, as one must be wondering that we have AI, we have everything, but who is going to control? Human, AI, I am talking about the last one, or the hybrid model. it's always going to be the hybrid model. See, there are so many technologies have shaped up in the world, but so far, do you think that human intervention is not required? Human intervention is required. That's why it's called artificial intelligence. Human intervention will always be required. So there is nothing that stops. Otherwise, that's the most important thing. So it will be always a hybrid model. Machine cannot do everything. Whatever data you process, whatever you do, after all it comes to a person's table. It comes to me, it comes to my computer, and I have to think whether I should use it or not use it. That's where hybrid model is always desirable. And there is one more term known as DMTA. You should pay a little attention to this. This is design, make, test and analysis cycle. This is also digitized now in the pharma industry, whereby this will optimize your process. Say for example, you use a reaction which is run at say 200 degrees with 20 ml of solvent. So this AI, generative AI will tell you how you are going to optimize this. What is the temperature you require? If you require only 190, why you are giving 200? Because I told you, impurity formation happens at higher temperature. If you use lower temperature, impurity formation will be less. Those kind of things, they are very important for formulator also and process optimization also. And quality control is at every stage. When you do some technology intervention, I told you, I gave you an example of weighing balance. So how do we ensure that weighing balance is completely qualified to give the accurate results? So that's where this AI tool will make an intervention. It will tell you if your product is not desirable, it will stop. It will tell you stop, you check this step is not okay, and then it will come to the next step only if you take care of the quality control, which symbolizes that there is a failure or this is out of control or out of spec. See, one more thing is drug development through GI. This has already happened. See, I told you 20,000 molecules are screened in the discovery stage. And when you come to animal studies, there are only 5 to 6 molecules which come to phase 1 trials out of 20,000. But when you have GAI, generative AI, probably if you start with 20 or 100, you may have 2 or 3 success stories. So that is how it is helping development of drugs in certain areas. Because if you give a simple command to AI that okay, I have ropicoxib, which is causing serious impact. What is the generative structure that I am going to propose to my research without having that cardiovascular impact? They will give an optimized process also. That because of this chemical, you are generating an impurity which is causing serious reaction on a prolonged use. So those kind of things can be taken care of this generative AI. So what is this? 90% of trials fail. Because if you have 20,000 and one reaches to the last stage and then it fails, success rate is also like 4 compounds going into phase 3, only one has a probability to pass. So if you take care of certain things in discovery stage, what I told you, to 20,000 instead of 20,000, if you do 200 molecules only through generative AI, you don't need to screen more, because this is very specific. They will give you 200 molecules, you may end up in doing 5 in a span of, not in span of 15 years, maybe suppose you have to screen 20,000 molecules, it's time consumption. 200 molecules is 1 or 2 years. So within, it will short span the time also, it will reduce the cost also. There is one example of one company. They started to develop one compound in 2019. In silico medicine demonstrated power of AI by creating a molecule. This is the name of the molecule because they don't disclose. This is for fibrosis. Just in 46 days. Imagine a thing that takes 2 to 3 years. This is developed in just 46 days. And this drug, if you even Google it, this is in phase 2 now. Very successfully passed phase 0 and phase 1. In 2020 itself, almost 500 clinical trials are conducted for the molecules which are based on AI/ML technology, where AI/ML has given a prediction in the discovery stage, and you have 500 molecules now in clinical trial, and this time span is also very short. Can you just move the slide? Next slide. This is smart manufacturing. Here also you can use the process automation using AI technology. Say for example dispensing, labeling, packaging, where human error is quite possible. Say if we have one drug paracetamol, and if we have similar manufacturing going on in other units, actually from a GMP standpoint that doesn't happen, but there is still a possibility of 1% of human error anywhere. Sometimes with labeling, sometimes with manufacturing dates, sometimes expiry dates, batches and all. Even that smallest error can be eliminated if we use the process and smart manufacturing automation. There are, based on AI, there are certain modules which are available in the market, or companies have developed their own modules of process optimization, quality control, but based on AI technology. So they are using this. There is one more thing, Internet of Things. So this is actually if you are doing multiple things in one lab. So they can connect the devices with each other so that you have a better control. Say I am a scientist, I am a formulator, I am doing something and my other colleague is doing something with a different device. So these devices can be clubbed together so that you have an integrated report and data. So again, this is very helpful. Have you seen some patients who are wearing the wristwatch like thing and monitoring the glucose? And they have a set of device here, so they can monitor glucose. If I take food at 2 o'clock, so 4 o'clock, this buzzer will speak that your glucose has gone up beyond 140 and up. Those kind of devices also are connected through IoT only. Heart rate, BP, remotely. Suppose I tell my family, I am busy in the meeting, please see the monitor at my home and then buzz me whether my glucose is okay. I want to take 2 gulab jamun today, my glucose is 90 only, so even if it shoots to 140, it's okay. I can have a relaxation of having sweet once in a week or twice in a week. So those kind of, that's a small example, but those kind of things, devices can be connected. This is a small example. These things are helpful to Internet of Things. They call it, this is a small definition. Then data analytics to speed biotechnology innovation. This is I am telling you, one big thing, gene editing has happened by the invention of CRISPR CS9. CRISPR CS9 is that is a bacterial enzyme actually. When there is an infection, so there is a process in the body which happens. So some of the, even gene editing has, I showed you first 4 drugs, all Nobel Prize winners. Then I showed you technological intervention of AI, 2 Nobel Prizes. Gene editing has also 2 Nobel Prizes. This was actually granted in 2020. So this is, if you have a sequence, protein sequence, you can actually chop the DNA and chop the mutation gene, so that if that gene is creating mutation in the human body at a later stage, you can make a protein without that mutation. That is what is gene editing through CRISPR CS9 technology. Then there is natural language process, communication between humans and computers. How do we communicate? Suppose I have a data. How do I communicate with the computer? What is the interpretation? Because there is a lot to do with the interpretation also. If I choose a wrong sequence and expect a good result, it will not happen. So for that, NLP is one more tool that can help the interface between humans and computers. Big data analytics to enhance insights to improve decision making. So again, this is all when we are dealing with a high class technology like this, which is just in the infancy stage, which is coming up, which is booming up. We need to train the human minds also. Then somebody should not think that human mind is not required. Human mind is very much required. Computer cannot do everything. That's why these additional tools are also there. NPL is there, IoT is there, then data analytics, this decision making is there. See, when you talk about decision making, that means human intervention is very much required for this. Can you go to the next slide, please? Precision medicine is always there is also, this is also catching up with the technology. This is actually when we make a medicine, say for example paracetamol. I told you we did a clinical trial on 10,000 patients, and there is a population of 6 billion people who are using Dolo type of drugs every day for headache, for fever and all. So precision medicine is whether this medicine which is made for 10,000 people, how can you give it to the whole population? I am not talking about paracetamol because that's a very old drug, new drug when it comes. So what they are trying to do is with precision medicine, that they will use the quantum computing and design a personalized therapy for a particular section of society or a group of patients who have a particular disease. So that particular disease, if they have only 60,000 patients or 1 million patients, that drug will cater only for 1 million patients. You can't give it to a general population. So digital prescriptions, you know, telemedicine and all, probably that's very common. Telemedicine, digital prescriptions and all. That's also one thing. That's a very small thing, but it is catching up actually. We have seen in COVID also that a lot of people are consulting the doctors on, and prescription is also coming, and then even to that extent now the prescription will go to the pharmacy. That happens in US. It goes to pharmacy people, and pharmacy people will deliver. They will give the training how to take the medicine. They will give a small box also. So those kind of things are happening with digital transformation. And even I would say that the service providers for these kind of services are from India only. There are certain industries in India which are catering the digital prescription, digital data handling for UK, US, Europe from India, because labor is cheap here. There is no other reason, because labor is cheap. And they are not paying so much of money to people who are working throughout the night to call the patient, how are you feeling, marketing surveillance and all. All these are happening through digital platform only. See, they are giving you some kind of, they have a template or they have a computer or a mobile, it gives you an alert. Have you taken the medicine? Yes. What's the feeling? Is your temperature gone down? Is your headache has reduced or not? Those kind of things are also digitized now, so that marketing surveillance becomes little easier. Now this blockchain supply, this is also very important. Blockchain, why we are using this? Do you know when the drug starts from a company, they seal the drug, they pack it. Now a drug has been manufactured in Hyderabad, it has to go to, say, Delhi. It has to go either by train or by a truck or a lorry. Do you know what happens in the way? There are so many other players also who are manufacturing spurious drugs. They are changing the carton in between. Who is taking care of that? This happens worldwide, I am telling you. Probably you may know, you may not know. Original drug is being taken away from the lorry because there is a big mafia working around it. Everybody knows this is happening. So how do you protect that? So you have QR scans, you have labels which are only specific labels which can be read by only certain kind of devices which everybody doesn't have. Suppose they have 3 devices on the way, and if I am not able to match the QR code on this device, which is control device, so that's how you can protect that counterfeiting of drugs and all. That's where blockchain technology in supply chain management is very, very important. this is a very, very critical thing that is happening in US and other countries. And we had 2-3 years back, we had a meeting with Consul General of US. She said this is the most important thing that is going to happen in the pharmaceutical company, because everybody was there, even Indian manufacturer. Because see, I am a person, say for example from Sun Pharma. I want to give the patients the best quality drug, but it's not reaching. It's not reaching to the patient. I am getting a superior drug with same label, same manufacturing dates, everything same, but drug is superior. So how do you control that? That this technology is going to help us out. Virtual training and simulations, I told you, this is again human and computer interface. Next please. This is a small example. How can we do this? The above process, we have 4 different sections where 4 different things are happening. Now this is known as continuous manufacturing. They are doing everything in one go. So everything in one go, process has started. You can say in one room everything happens till your tablet is manufactured. That is continuous process. And there are process control designs, QBDs, taken care, quality control, regulatory perspective also. Because see, I will give you 6 medicines have been so far produced by top companies using PCM technology. Process of continuous manufacturing. I'll tell you about regulatory also. I want to tell you one thing. If I ask you a question, whether science is above all or regulatory is above all, you tell me. Who is at the top? Anyone? Why? See, science comes first. See, regulatory people, they learn from science only. I told you, when you do mistakes, and then only regulatory comes in picture. So science is always on the top. You should always remember this, because see, regulatory people tell you do. They are not stopping you. I gave you one more example, like if there is a trial happening on 5,000 patients. Regulatory tells you, okay, you have spent so much of money, you market it, but do a post-marketing surveillance. We are interested in post-marketing surveillance, whether this drug is benefiting the population or not. That's why science comes at the top. So it's our responsibility as scientists to do a good job at our bench, write everything, keep in mind that data integrity is very important. Whatever work I am doing is going to benefit the society. See, somebody may be contributing millions of rupees to society for feeding poor and all. That's also one kind of seva we are doing. Do you know what is the best seva in this world? Feeding a poor or what? Can I tell you? The best seva, Nishkam, seva is enabling somebody through wisdom. That is what Swami Vivekananda told in Chicago. If we enable somebody through our intellectual learning, wisdom, that's the best Nishkam karma. It has highest place in the world. So that means if we are scientists, if we are doing something, we should do for the cause of human being. We should leave everything else. No negativity should come in our mind. We should do our best. If I do like this, my other colleague will also like that. So everything will fall in place then. So that's the message I am trying to give. Next please. So this is also 3D printing. Do you know what happens? So they are putting layers of drugs like this. It's not mixing now, no more mixing. They are just putting the 3D printing like this, and I have shown you the cyclized release control. There is some material like nanoparticle and all. They are putting it on the layer by layer, so it becomes easy. 4 tablets in one. 4 tablets, 4 medicines in one tablet. Because for geriatric patients, for children, how can we remember to take 4 medication? At the age of 40, if somebody is taking blood pressure medicine, then diabetes also comes in, then Disprin also comes in, aspirin. So typically by the time we reach age of 60 or 70, we have 5-6 drugs, morning 5, evening 5. So this technology is very simple, and they are using simple technology 3D. And there is one drug already which is in the market, which is approved by 3D. Last, I think we are on the last one. This is personal pills, polypills, chewable, control release. This is all application. See, you just remember this. There will be a lot of information available on the internet. Can you go to the next slide, please? I think we have now last slide. Gene therapy, I already told you, there is CRISPR-Cas. This has won a chemistry Nobel Prize in 2020. You can modify certain genes so that you have a better sequence, so that you can develop a biotech drug which is not causing the gene mutation. So sickle cell anemia is one drug which is already approved in UK in December 23. This is by this technology. So it is already available in the market. And then there is one more CAR T cell therapy. This is by Gilead. I told you, go to Gilead's website, you will find a lot of information there. 4,000 patients. This is again what I was telling you, personalized medicine. This is the example of personalized medicine. Can you go to next please? Thank you for your attention. And if you really have to see where you can make an impact, can you go to next slide please for opportunities? Just one slide. Oh, it's not there. Okay. Have a nice time. I wish you all the best.
Research. We are extremely obliged for being part of the session. Thank you, sir. Now, I take the pleasure of inviting Dr. Arunabha Malik to honor our guest with memento and shawl. I request all the doctorates to join for the ceremony.
So we are extremely sorry to interrupt you between the session because of the delay in the lunch and all. Students are also sorry, sir, and thank you so much. Please come.
experts representing diverse domains across drug development, clinical research, regulatory science, and industrial pharmacy. Their collective knowledge and dialogue promise to spark thought-provoking conversations and actionable insights for the next generation of pharma professionals. We encourage our audience to actively engage during the question and answer session and make the most of this opportunity to interact with these eminent minds. Let the exchange of ideas begin.
It is my privilege to introduce one of our distinguished panel members for today's discussion, Mr. Srinivasulu Gunta, a highly accomplished pharmaceutical professional with over a decade of rich experience in formulation research and development. Mr. Gunta holds a master's degree in industrial pharmaceutical
It's my pleasure to introduce Ms. Anushree Saha, an accomplished clinical data management professional with over 7.5 years of industry experience. Currently working as analytical data manager professional with over 7.5 years of industry experience. Currently working as analytical data manager professional with over 7.5 years of industry experience. Currently working as analytical data manager professional currently working
That's the reason there is no. So, I would ask the simple question: why you have joined the B Pharmacy? See, people are laughing. So there is no answer. Okay, then fine. Okay, then leave it. So just want to do a degree and you came for graduation. I have to be some graduate, then I have to fly off some country, or I have to do for some government job like that, I guess. But I'll tell you, there is a lot of science. There is a lot of gap between the industry and the colleges. That's the main reason why people are they fear to get freshers into the industry. So you have to train yourself. You have to people are they're here to get freshers into the industry. You have to college. You have to any kind of difficulty. You have to train yourself. You have the same they're here to get freshers into the industry. You have to college. You have to any kind of difficulty. You have to train yourself. You have the same they're here to get freshers into the industry. You have to college. You have to any kind of difficulty. You have the same you have college, you have most of them are from B.Pharm here, right? With the bachelors. Most of them are from B.Pharm here, right? With the bachelors. Most of them are from B.Pharm here, right? College bachelors. Most of them are from B.Pharm here, right? Regulatory is there. Apart from we have a project manager. Regulatory is there. Apart from we have a project manager. So my best suggestion, if you want to do the regulatory, I want to just probably have a project manager. So my best suggestion is very well want to do the regulatory, I want to just probably have a project manager. So my best suggestion is very well want to do the regulatory, I want to just probably have a project manager. My best suggestion is very well want to do a lot of regulatory, I want to just probably have a project manager. Anyone saying I want to become a scientist, still there is an ambiguity. So, okay, I'll just leave it. So you have any particular question for me so that I can answer you? No questions? Okay. Then I'll give a chance to other panel members, they'll explain you in detail. Okay? Thank you so much for this opportunity.
I now request Mr. Vikrant Kari to please take the session.
Hi, hello everyone. So I think sir is from the same department which I am working. only his expertise into the tablet, but I am expertise in oral liquids and topical formulations. I think he explained already all the process in formulation development, so I don't want to repeat that. And I think you people are not in mood to listen also. So, so I'll just explain like topical formulations, what are different cream, gel, lotions, and in topical again cosmetics also comes in topical formulation. So maybe some ladies are interested in cosmetic formulation development. So maybe I think I don't have anything new to tell other than what sir has told. So if you have any question, you can ask me, or else the basic things in topicals that I can explain. Like in topical formulations, generally it is cream or ointment. So creams are oil in water emulsions, and ointments are water in oil emulsions. So maybe in the pharmacy emulsion, maybe the one of the topic in your syllabus. So you can tell me what is like difference between emulsions, these two type of emulsions, if you anybody have interest to tell. Okay. So I think the process of development of tablet dosage form or any other dosage form, the process is same. It will always like selection of API, then selection of excipient, then taking trials into the formulation R&D, then finalizing formula, then loading that formula into the stability, and then taking the quality parameters in analytical development part. And once stability is done, then scale up batches and process validation batches we need to take, and then commercialization. This is the common process for all the dosage formulation development. Only is like critical parameters or critical steps involved in OSD and other dosage form is different. Like in oral liquid formulation, just if you can say in oral liquid generally cup syrups or for formulations required for kids, that is goes into the oral liquid formulation. So when you are planning to develop any product for kids, so you need to take care of flavor of that formulation. So that is one of the main criteria to mask the taste of any APIs. And if you people like know whatever the cup syrups we have in market, so all the APIs used in cup syrup, all having bitter in taste. So if you check any cup syrup, you will get bitter taste only. So masking that taste, then masking the flavor, giving some additional flavors, that is one of the main criteria in development of oral liquids for kids formulation. Then solubility of API, so during their stability it should not get precipitate out. So that like few critical parameters in the development part of like oral liquids. In topicals we can see like phase separation when we are formulating any emulsion. So during stability or at higher temperature, formulation gets separated, oil will get separate from the cream. So that part we need to take care during the development part. So selection of emulsifiers, solubility of emulsifier, concentration of emulsifiers plays the main role in this stabilization of topical formulation. Maybe like customer wise, if you see when you are taking any tablet from the market, if you find that that is broken, generally you will not find any tablet that is broken. But you don't know whether the API is present in that tablet is correctly or not, whatever like 100% it is there or not. You people consume that as it is. But in topical formulation, issue is when you will buy any cream from formulation and if you find out oil is coming out from the cream, so anybody can understand there is some problem in this formulation. So that is the main like quality affecting parameter which can understand by any patient. So that is the main parameter we need to take care when you are developing any topical formulation. That phase separation should not be there. So this is like some basic things which I am able to remember to convey you. So now if you people have anything question, you can ask me. Okay, thank you. Thank you for all this opportunity.
May I now request Ms. Alushri Sano.
Hello everyone. Good afternoon. I hope everyone is not feeling sleepy. Am I audible to everyone, to the last people? Yeah. Am I visible because I'm short? Okay, fine. Okay. So I have 7.5 years in clinical data management. So does anyone know what is clinical data management? Any slight idea, anything? Anyone aware? No one? Okay, fine. So even when I was studying pharmacy, when I finished my masters, I was clueless as what to do. So it was coming, like data management is coming. So it was very new when I passed out from my pharmacy degree. So when I got into this industry, I got to know a whole world is there which is related to data. So data is everything right now, okay? So you're born, you have a birth certificate, that is data. You are getting married, you have a document that is proving you are married, you are not single. So that every document is a data. So my job is to make sure that the data is validated and clean. So before I go ahead to data, so everyone has seen, we have gone through the COVID, very pathetic situation. It's a completely new disease, right? So we got a new vaccine like Covishield, okay? So how come we got to know that like this vaccine or this drug is going to work? What happens from identifying the disease till the drug is being released? What happens in between? Does anybody know? Sorry? Trials? Yes? Yes, correct. So clinical trials, correct. So when a new drug, when a new disease comes up or in a pre-existing disease, so a new drug or a combination drug is developed. So we have experts here who have already explained different formulations. So once a new drug is identified, a new molecule, a new vaccine is going to come into the picture. So a series of clinical trial happens. Okay. So when the clinical trial is being done, so we get a bunch of data. So because during the clinical trials, a number of assessments are done, some safety, efficacy, all this information, the side effects, the serious adverse events, the dosage, everything is determined, everything is documented. Because we need them documented for future purposes so that once they are good, for example, a drug like Covaxin and Covishield, we have it. So they must have seen this drug or this vaccine is actually working. So when they make sure that yes, this drug is actually working, it needs to come into the market. So before going into the market, we get the data, and it's our job as data managers to make sure the data is validated. So that is during the clinical trials, number of patients are coming, a lot of tests are happening. So all this data are collected into certain databases. So we have multiple databases. I am working in something called RAVE, while some other companies may have Inform. So you will hear all these terms once you get into our industry. So these are the databases that are used to collect that information. Whatever tests and analysis is done, the pharmacokinetic information, the antibody information, any sort of information that is getting collected, they are documented in those databases. And now it comes to us. We are the ones who clean the data, who make sure everything is crisp, everything is perfect, so that these go ahead, so that the drug or the vaccine, whatever is coming into the picture, these go ahead to our regulatory authorities and it gets passed and it comes into the market. So that it comes to us and we become the end users of that drug. So that is a basic and layman language what I wanted to express to you guys. So we have, actually you are in the right place at the right time. We have multiple companies right now in working with clinical data management. Right now in Sanofi, you have BMS, you have Novartis. I think we also have a lady with us, from Novartis. It's like a hub opening up. So if you are interested, then definitely come into the picture. We will be welcoming you with open arms. And there's no hard and fast rule that you have to complete your M.Pharm or you have to do M.Pharm to get into our department. You can complete your bachelor's degree, and if you are interested, definitely we are going to welcome you. So thank you all. Thank you for this opportunity, and hope I did not bore you guys. Bye.
Thank you, ma'am. I request Ms. Vanita Neti to please take this.
Thank you, ma'am. Sleepy already? Yes, good morning. Okay. Okay, good afternoon all. Thank you for this invitation, and yeah, glad to meet you all and glad to see you all. Special thanks to Dr. Harita, my dear friend. Yes, I'm from regulatory affairs team department, and how many of you are aware what is regulatory affairs department do? Any knowledge? Any overview? No, okay. So let's discuss about lab to life, okay? So masterminds like you all work in the lab, and we regulatory team come into the life, yes. So all the masterminds work in the behind the screen to develop a drug, and then regulatory team comes into the picture. Regulatory team speaks with the health authorities like US FDA or EMA. They are called as regulatory bodies, like all the expertise who have already mentioned. And they are the ones who evaluate our development, and they are the ones who approve our drug, and they are the ones who ensure that our medicine is reached to the patients who are in need. You might already be aware of the regulatory bodies like US FDA, EMA, and so on so. And you also might have heard that US FDA has inspected so and so company, and EMA has audited so and so company, and EMA has or US FDA has noticed a few observations in so and so company, right? Yes. So once a drug is developed, a regulatory team will negotiate with health authorities for getting approval for that product, and finally we see to that we ensure that the drug is approved by the respective health authorities, which are called as FDA or EMA. If it is approved, then our hard work is fruitful. If it is not approved, then what? Then that is again different. Why is it not approved? It's because FDA or US FDA or any health authorities, patient matters for them, right? Patient health matters for them. And if we are not manufacturing any product with required standards or qualities, definitely we are not, the drug is not going to be approved. And the most important thing is we negotiate with health authorities, but it's not very simple that they ask for many justifications, many data, like what our expertise has already just made us aware. Yes, we have to provide them lots of data, and there are also chances like they visit the companies, they audit the companies, they inspect the companies, right? So you might be aware, like you might be also seeing in the news channels and newspapers. Yes, we should be very ethical. We should ensure patient safety, quality of the drug, everything. Once the product is approved, then it is launched into the market where it is actually required. If it is in US, then US FDA approves the drug, and then there it is launched. And it's then the picture is not finished there itself, like it's launched and it's over. It's not like that. We should also maintain it. Maintain in the sense it's in the market, we should keep on ensuring its safety. Initially we manufacture the drug or medicine with proper standards, and then it's already into the market and it's already established, then we can compromise with the quality of the product. It's not like that. Regulatory authorities, they keep on monitoring the drug, they keep on ensuring that the safe drugs are reached to the patients. So if they found out that something is going wrong, definitely they reach out to the companies, and we are always answerable to them. So the key point is we need to maintain safety, efficacy and quality of the drug throughout it is available in the market. And if not, what happens? Just imagine a common drug like paracetamol is not available in the market. Just imagine, because we all are very much dependent on paracetamol, right? For headache or fever, whatever, we immediately go to the pharmacy, even without prescription. We just take paracetamol and take it. Just imagine there is no paracetamol at all.
just because of compromising its quality. Can we imagine? No, right? Yes. So quality of medicines is important. If medicine is not available in the market, then definitely patient's health is affected. Our key point, whoever, like wherever we are, in whichever department we are, our end point is definitely patient, right? Directly or indirectly, we are impacting the patient. So whatever you do, whatever you learn, explore yourself and always maintain your standards or quality. Yes, we all together can definitely build a healthier world, and we all can fight situations like COVID if we are upgrading ourselves. Yes? Any doubts? If you wanted to ask something about regulatory? Last? Okay. Thank you all.
Thank you, ma'am. I request Mr. Kishore Kumar, please.
Good evening, everybody. Thank you for giving this opportunity. My senior madam, Sri Harita madam, sorry. Okay. Actually, I'll say one story about the dosage forms. When I am traveling into my village, on that time, my children continuously vomiting. And that time, my wife, you are the pharmacist, what you do? Simply say, she is coming from background of BTech. So I thought, okay, traveling, on the traveling, I take one strip from the medical shop. Strip, you know, strip, oral disintegration strips. Ondansetron I took, then give it to my children. Immediately vomiting stopped. She happy, my wife will happy. Why? Because you are a mantrika. Yes, I am really mantrika. So I introduced one new dosage form about that is called as a oral disintegration film. Are you listen about the tablets, capsules, syrups, lotions, emulsions and injectables? But in the academics, oral disintegration films are not in there, I think. So in this oral disintegration films, having onset of action is more. So conventional oral doses forms, tablets, capsules face many challenges delivery systems. Onset of action it is difficult. So we will take a oral disintegration films. Next slide. ODF is the thin, flexible, non-brittle postage stamp size strips included one or more active pharmaceutical ingredient, dissolve, disintegrate with a few seconds, saliva, without water or with water also. The sublingual mucosa has a high membrane permeability due to its thin membrane structure and high vascularization. So that's what onset of action more. Next slide. So saliva contact, then fragment, then active ingredient release and base separated, action will be started. Okay, next slide. Advantages of ODF: easy to apply, no risk of suffocation, suitable for pediatrics, geriatrics and bedi patients, provides a rapid onset of action, conditions required urgent intervention in an allergic attack such as asthma and vomiting, bypass the gastrointestinal tract and thus increasing the bioavailability. So these are the more advantages of oral disintegration films. Bioavailability equivalent to oral disintegration tablets also. Next slide. So a simple formula, you know, tablet having a plenty of steps, injectables also having a critical parameters. but in the ODF, very less ingredients and very less procedure. So in that formulation we have take API and plasticizers, saliva stimulants, sweeteners, super disintegrants, flavoring agents, surfactants and coloring agents. Next. So this is the flowchart of formulation. First we have to prepare the formulation in what? First we have to prepare the polymer into water, that is called as the base, and disperse it into the excipients and homogenization and layering, drying, slitting and packing. Very simple procedure and flowchart. Next. Next slide. This is the machineries available in the industries. This is a new technology and novel technology. So that is called as a dryer. When we are pouring into the one side, we are pouring the slurry. Then next side collect the strips, mother roll. That mother roll cut it into slitter rolls. Slitter rolls into cut it into what we need the size. Next. Evaluation parameters are visual inspection, thickness measurement, pH, moisture and uptake, moisture content, assay and dissolution, disintegrations. Next. Limitation. Not suitable for drugs that cause irritation in the oral pH. So, okay. Not suitable for higher doses. Hygroscopic nature APIs also not suitable for this formulation. Okay, next. Examples of ODF in global market. Nutraceuticals also available, vitamins, minerals, all the things. Next. Thank you for upcoming entrepreneurs. I look around this room filled with bright minds. I see not just future pharmacists, I see a future scientists, technologists and policy makers and healthcare leaders. Okay, thank you.
Okay, we are opening the session for Q&A session. I request all the students to please brainstorm any questions related to the current field, current trends, anything as such, and you can ask our expertise there. If you can raise your hand, we will hand over the mic to you all.
Location.
Good afternoon everyone, myself Anurag from B Pharmacy. So my question is regarding clinical data management. So what is the primary role of a clinical data manager in clinical trials?
Hello? Okay, so the primary role is a vast role, to be honest. But the basics of it is to make sure the data is accurate. So whatever the data is getting collected in the databases, we make sure everything is up to date, nothing is missing, and everything is as per compliance. So that because once the data is accurate and validated, it will go to for the FDA approval or EMA approval. So that finally the drug comes into the market. So our role is to make sure the data is perfect and there are no discrepancies identified.
Thank you.
Good afternoon, everyone. I'm Chaitanya from B Pharmacy. So my question regarding clinical data and management. So can you explain the role of clinical data manager in clinical research and therapy?
Hello. Okay. As I previously mentioned, so our flow is the data. That's the main part. So, for example, as I was in part of one study you mentioned, ICARIA study. It was related to a drug called isotretinoin. So it was a drug for multiple myeloma. Before the drug came into the picture, and it got an EMA approval actually in the year 2021, I think. So I was part of this study. So it was my biggest, I would say, achievement that I was part of this study. And in there, we actually each and every data that we have, that we received from the site, we made sure that it's actually it's compliant for the FDA approval or EMA approval. So yeah, it's actually our main role is to play with the data, to make sure the database is accurate, make sure the instructions provided to the site is actually correct. Because basis on our instructions, site does the data entry into the database. And we also help the data, help the sites to, we train the site, help the site to understand the process of the data entry and also understand the queries. We are raising multiple queries to the site, asking to correct this, correct that, make sure the unit is perfect. We are it's an integrities of each and every data. It's not only just this one point or this one maybe a number or one unit. It's like each and every information has to be validated, has to be accurate, and it's like you will be, once you get into the industry, you will see so much of challenges that you're going to face in them. And we have multiple tools in Sanofi which help us with the process. It's not a manual work. That is one good thing that we have. We do not have to do much manual work. We have multiple tools which help us define the discrepancies, and we look into the discrepancies, and then we take the appropriate action.
Good afternoon, ma'am. Good afternoon to everyone. Myself, Varshita from PharmD first year. So my question is regarding clinical data management. Ma'am, you completed your pharmacy degree, so you had a complete idea about pharmacy. But how did you understand data management? How did you choose this career-wise? And what you had to do, like which degree did you do so that the companies will accept you in the field of clinical data management?
To be honest, I had no idea what it was data management when I passed out from my college. It was just by chance I came into this industry. Cognizant was the one who did, I was also it was a college campusing that I was hired. So I was doing my masters in pharmaceutics, but it was not restricted to masters also. Even after bachelor's degree, you can apply for the job. Data management does not have any, it's very stringent educational criteria. So even after your bachelor's degree, you can come into the industry. So yeah, I don't know whether it's there in your syllabus in your bachelor's degree, but in our masters we had one chapter for clinical data management. We had a little bit of idea of what it is, but once you get into the industry, you will get exposure to multiple portfolios. It's not only data management. Data management also consists of programmers, developers, statisticians. We work with everyone. And it's one of our key role is to communicating with the clients, communicating with the sites. So it's not only data managing, it's also communication that need to be a part of your factors.
Thank you, ma'am.
Thank you. Anyone else?
Good afternoon, everyone. Myself Surisha from Ecom Final Year. So I have a question regarding regulatory affairs. So can you give an example of complex regulatory issue that you have helped to solve?
Hi. Very confidential question you have asked. Okay. So I cannot share the real time thing what I have done, but maybe I can make you aware of something which is general. Maybe if you see COVID medicine, generally for drug to get into the market or to get approval, you might be aware that it takes years of time and it depends upon the innovator or generic. For generics, it takes less time to get approval, but for innovator, it takes years of time to get approval and to make it available for patients, right? In case of COVID, you have seen we have taken vaccines within very less span of time, yes, and they are all innovators. They are not generics and they are innovators. So in that case, regulatory team comes into the picture where it's a very complex issue. They have to negotiate many a times with health authorities. They have to speak with them and they have to provide justifications to health authorities, which is really a challenging thing, and they have to plan lot many strategies because all the technical terms, whoever develop medicine, they will be knowing like what's the behind screen everything. Only developers know that. But regulatory team should at that moment has to pitch in. They should try to understand what health authorities are asking, and they should immediately plan the strategy, and they should give commitment to the health authorities that whatever they are asking for, we will provide at so and so time. Those commitments, and see to that the product is launched on time in market and made available to the patient. Okay, so this is something complex I feel, because making drug available to patient is not a simple thing, that too in very crucial situation like COVID. Okay, so that's something complex, which is in general I wanted to tell.
The basic complex thing that triggers in the drug development is the development. If a formulator or if the scientists do the good development, the regulatory people or other people, they won't face anything. Even the agency won't get any problem. Even the CDME or the clinical studies, they don't get any issues over there. The main criteria and the main this thing is the basic development. The basic knowledge lies with the scientists who work on the drug development. That anything that gets into the market will start from the drug development. So the basic science is involved in this. The scientist who works on this should be more efficient enough that the developed product should not get any complication in future, that it's stuck into the regulatory part or it's stuck into the agency. I mean to say whether it's an FDA or it's EMA or even we call it as CDS, it depends on the country. So the basic thing what I mean to say is that you should have to train up on the subject what you are studying in the bachelor's and as well as in the master's. That helps to become a good scientist. And that initiation will resolve many problems, even in regulatory, even in CDMO, even in the clinical studies, even in the pilot studies. So this is that's I want to say on you guys.
My name is Anusha from Guru Nanak Institutions of Pharmacy, second year. So my question is, what is the use of max in pharmacy?
So you tell me one area where maths is not used.
Sir?
You tell me one area where maths is not used.
Sir, for calculation is another thing. What is the use of matrix, integration, differentiation? What is the use of that?
Okay. That's very good question. You should have a clarity on this. Okay. So basically maths in terms we just starts with the numbers. So for example, in the drug development, everything that in some one of my person has shown you some specification, which is they have some dissolutions, disintegration tests. Anybody know what is dissolution?
We don't know, but we need specific reason why we need maths for the pharmacy. This type of things like matrix.
That's what I'm saying. That's what I'm saying. You know what is essay?
Yes, sir.
What is essay?
Essay in the sense writing.
No.
We can, sir, in matrix we won't learn about essays, no sir.
We just give it. You have seen dosage form. There is a 6 mg, 20 mg, 40 mg.
Sir, okay sir, accepted. What about trigonometry, sir?
I'm not doing trigonometry in drug development. I'm not using trigonometry.
So this is what I want to ask, sir.
So why you got this question? Like why trigonometry?
Sir, because we are BPC students, no sir. It is critical to read that MPC, you know sir. Maths is so hard for us. The reason I got a question on this. So there is no need of maths, no sir. So the reason I asked this question.
No, without maths, how will you do science?
So it should be like basics, sir, not critical questions like matrix, integration, trigonometry, etc.
I got it. Don't worry about the complex mathematics, okay? We do develop complex drugs. We do not develop complex mathematics.
Thank you, sir.
I'll answer. Yeah, I'll answer.
You'll answer the question? Oh, wonderful.
Yeah, can I say something related to mathematics?
Yes,
So, pharmacy, how you feel that chemistry is very, very important. Similarly, mathematics also very, very important. Not only for the calculation, making calculation for the formulation and making percentage purity, all those things. Whatever the laboratory experiment you do, the mathematics are involved. Apart from that, later when you do your research, those mathematics, whatever you have been mentioned, that is very, very important for the data analysis. When you go for the statistical analysis to check whether the significance effect of any kind of drug molecules against particular when you do the biological activities. So by the time it will be very useful. So maybe you might be an undergraduate student, so you might be thinking you may not apply much in the theory knowledge in the practical. But later when you go for the masters or PhD level, when you do the research related to any kind of field that what the industry feels, so definitely you need to apply all those skills. So don't think that is not important in pharmacy. So later anyway it is important if you go for postgraduate level when you do your research, especially for the data analysis and statistical analysis. You need to know more softwares. Okay, so those softwares also you need to know some knowledge about mathematics. So that is the main reason that may be included in the syllabus as well. Okay? Yeah.
There won't be any trigonometry or like this thing. There won't be any, this. But there is basic mathematics you should have, like calculations or permutation and perturbation. It will help you. In the drug development, you do drug by design. The design involves mathematics, but not trigonometry.
Trigonometry also is required. So when there is a product, if you want to characterize the product, so lot of things will come into the picture. Maths, physics, chemistry, biology, all together is required to develop a product and characterization of the product. So trigonometry will involve during the characterization of the product where crystallography comes into the picture.
When you want to actually get a picture of how those bonds are bound, in what angles, there your trigonometry will be coming into picture, right? Matrix, so answer for everything. Yeah. Actually, your BBPK teacher in your 4th year, you are in 2nd year form, you know. So BBPK, you will get answer to all of your calculations. So don't worry. So there is a lot to learn. Your formulations in your 3rd year. Madam will answer you. Madam will answer you the entire.
Over studies will be there. Under that and over studies, unless otherwise you are performing all this mathematics, you can't. That much importance is there. When you are going for the 4th year, you can get right. Those calculations, everything is all these learnings are to fill our pockets, right? To calculate or to break down our salary, also we need math, right? So yes, math is important.
Good evening, sir. I'm Sonu from KRN College of Pharmacy. So I have a couple of questions. My first question is for Kishore sir. Sir, you have said about ODF. Why it is not mentioned in our syllabus, sir? And it has so many capabilities of it is having onset of action. Quick onset of action in it just results in our mouth. And my second question is just general question, sir. It's for Mahendra sir.
One by one.
I want
Your good name?
Sonu sir.
Sonu. Actually, novel drug system is now modern developments now. That's what. So having a drawbacks of tablets, capsules, these overcomes to came the ODF. That's what not incorporated in your syllabus. In future, might the government will be put. Okay.
Thank you, sir. Sir, my question is, sir, yesterday you have said about AI, sir. So I have gotten a doubt for that. Is there any deviation or impact caused by the AI on future for our responsible pharmacists? Like if we take a trending example like ChatGPT, it has a Ghibli style ads. If we give a topic and a prompt, it will just generate an image in 5 to 10 minutes. And if a professional animator takes about 2 to 3 hours to do it, then how what is the guarantee to us to multinational companies hire just robotic AI bots and not us?
So AI, I think you know it is now every field I think that is take over everything I think. So in the pharmacy field, especially even yesterday during my presentation also I mentioned. So empathy is definitely AI cannot give the empathy. So when handling the patients, especially the doctors or pharmacists when counseling the patients, so that cannot be taken by the AI. AI is a tool that maybe we can implement in the main instruments, machine learning and deep learning, so which is going to take over for many things in the pharmacy. Okay, but because of empathy, definitely manpower is required. As a pharmacist, as a doctor, and in other field it is okay. Engineering kind of field, it's okay. We can implement AI tools which can reduce the manpower and reduce our workload. But still, even though AI take over everything, still manpower is required to do all those things. Okay.
So for if there is an analytical thing, then the companies will have this only the bots, sir. If it is no need of normal pharmacists, then how can we see that?
It is not something like that. It is going to happen, but not exactly what we are thinking right now. It's not going to reduce lots of manpower. So maybe slightly it may affect the manpower requirements in future. But the future will give the answer for all those kind of things. Because currently we don't know whatever the handphone that we are using. 10 years before we never been thought that we are going to have everything within our hand. You can check whatever you want, know everything. So something like that. In after 5 years or 10 years of time, by the time we come to know what is AI tool, that how it is going to take all the manpower related work and how the manpower is utilized to all those kind of things. So the time will answer for all those kind of things. So we need to wait for it. But definitely manpower is required for any kind of situation. Okay, but time to time maybe AI is still taken, but still manpower we have to give some kind of input to the AI tool as well.
Sir, if AI goes through, then what is the use of studying, sir? Because every drug is just defined as ChatGPT. If just added a prompt.
Yeah, even this is the problem. Recently I discussed with one of the faculty member also. Because previously, just 2 years before, 3 years before, when you ask the students to write an assignment and submit, they find very difficulties to write an assignment. But nowadays, if you ask them to write just 250 words or 500 words, they were able to make even 5 pages or 10 pages like that because of ChatGPT. So they can get lots and lots of information from the AI tool. Even the lab report can be generated. Just you can ask what are the questions from the ChatGPT, you can get answer from that. So that definitely affected even all the assessment as well. Even when we conduct the lab experiment, we usually include the pre-laboratory and post-laboratory questions for the students. But nowadays, that also easily they are getting answer from the ChatGPT. So it is the biggest task to all the lecturers making these kind of assessment, and we are now, especially in my university, so now we are focusing on how to fight with this AI tool and how to make better assessment to assess the student learning progress. Otherwise, definitely we cannot assess anything because everything they are taken from ChatGPT. And nowadays, what we are doing, even we are checking the some of the software by checking whether the students really write the real experience or that is generated from the AI tool. So that kind of restriction has come already. Even if you submit now article for publication, any manuscript or any written kind of things, nowadays we have a software that we can check whether that is originally created by the manpower or it is created by the AI tool. Okay, so such a way I think in future in academic also, you got a solution to how to make it. Similarly, the assessment by the lecturers or academicians, we also have some tool. We can identify whether the students written properly, it is a man-made or machine-made like that. So that such kind of things are there. So in future, in every field like that only. Whenever the AI is developed, there is another way how we can, whether we can detect the knowledge of students or the learning progress of the students. There are many things are coming out actually in the future years. Yeah, any more questions?
Thank you, students. May I now request Dr. Arnab Maligaru, along with the doctorates, to please come forward to raise our thanks.
ఏదో రాబాయ్ ఎట్లనో అట్లా చేసుకొని తీసుకురా సైలెంట్. పడుతుందిరా పడుతుంది, క్లిప్కి మధ్యలో పడుతుంది కదా. డాక్టర్ ఉంటారు యాడ్ చేసుకుంటారు నాకు అది అందరం నంబర్ నా దాంట్లో ఉంటుంది కదా, దాంట్లో
I request all the panelists to please step down and we will have a session.
With vibrant cultural performance followed by the glimpses of the best moments through our 2-day journey with all participants, delegates and our esteemed guests. Please welcome them.
Thank you.
a video of 2 days event.
అయిపోయిందా అరే తొందర చేయరా అందరూ వెయిట్ చేస్తారు ఏందా హలో వీడి నంబర్ ఉందా కొత్త
Good afternoon, everyone. As we approach the closing moments of the intellectually invigorating and inspiring journey over the last 2 days, it's my honor and privilege to welcome you all to the valedictory ceremony of the International Conference on Pharmacy 4.0, Revolutionizing Healthcare Through Innovation and Technology, organized by Marri Laxman Reddy Institute of Pharmacy, in collaboration with the Telangana State Branch of APTI. The conference has served as a remarkable platform for knowledge sharing, collaboration, and forward-thinking discussion on the transformation of pharmaceutical sciences in this digital era. To grace this occasion, may I have the honor of inviting our esteemed dignitaries to the dais. The visionary guiding this conference, Dr. Arunabha Malik, Convener and Principal, MLR Institute of Pharmacy. Our chief guest for the valedictory session, Dr. Sunita M. Reddy, Professor and Principal, JNTUH University College of Pharmaceutical Sciences, Sultanpur. I request all the dynamic doctorates to grace or to the dais. Thank you.
I request Dr. Mahindan Sekhar to please proceed.
We are truly delighted and deeply honored to have amidst us our chief guest for the valedictory session, Dr. Sunita M. Reddy, Professor and Principal, JNTU University College of Pharmaceutical Sciences, Sultanpur. Ma'am, your presence here is a source of great inspiration to all of us. With your immense contributions to pharmaceutical education and research and your unwavering commitment to academic excellence, your words today are sure to leave a lasting impact on every participant here. It is with great honor and admiration that I take this opportunity to present the profile of our esteemed chief guest for the valedictory session, Dr. Sunita M. Reddy, Professor and Principal, JNTU University College of Pharmaceutical Sciences, Sultanpur. Dr. Sunita Reddy is a distinguished academician, accomplished researcher, and an influential leader in pharmaceutical education with over 3 decades of professional experience spanning across India and the United States. She holds a PhD in pharmaceutical sciences and a master's in pharmaceutics, having earned a university gold medal from Kakatiya University from academic excellence. Currently serving as the principal at JNTU UCPSC, Dr. Reddy has previously held various key academic and administrative roles at the Center for Pharmaceutical Sciences, IST, JNTU, Hyderabad, including Head of Department, BOS Chairperson, Women Grievance Cell Coordinator, and Placement Officer. Her multifaceted contributions have significantly shaped pharmacy education and institutional development. Dr. Reddy's extensive experience also includes her work in clinical pharmacy in the USA and her leadership in setting up diploma programs, research collaborations, and numerous skill development initiatives. She has successfully guided more than 170 M-Pharmacy students and 14 PhD scholars and has over 170 research publications and multiple book chapters to her credit. Her exemplary service has been recognized with numerous prestigious awards such as the Best Teacher Award from the Government of Telangana, the Women Excellence Award from BioLeague International, and the Pharmacy Teacher of the Year award from the Pharmacy Teachers Trust, among others. She is an active member of several academic councils and governing bodies across universities and institutions, and has played an instrumental role in organizing over 80 workshops and conferences, further fostering academic and industry collaboration. With a strong commitment to innovation, ethics, and empowerment, Dr. Sunita Reddy continues to be a beacon of inspiration in the field of pharmaceutical sciences. We are deeply privileged to have her with us today. May I now invite Dr. Sunita M. Reddy to address this august gathering.
Good evening, everyone. Okay, are you all here or are you thinking about your buses? Okay, good evening, everyone, and thank you, Akshita, for introducing me. People they call after 3 decades, they are veterans, isn't it? So I am one of them. I have been working from 91 to till now in different things, and still I am working. I have to work 7 years. Okay. All this is fine, but you know, I might have been very, very happy if they had called me for a talk. Okay, I'm just kidding. He already told me that, because revolutionizing healthcare through innovation and technology, that is one of the favorite subjects for me, because usually wherever I go, like I go like in Thailand, Malaysia, and Singapore, US, Chicago, somewhere, we talk about that. We do a lot of workshops under innovation and technology, and especially nowadays, the healthcare system is coming and revolutionizing with the AI, okay, artificial intelligence. And good evening to Principal Marri, Dr. Marri, and all colleagues of the MLRITM, because I do not remember few names, and also the guest from Malaysia, and my dear teachers, and my dear students. Please feel proud, because we are all pharmacists. Okay? That is the thing, and we try to see a satisfying thing, that because we are a pharmacist and we are working in innovations, and we are going to give either new product, new molecule, or new something to the society, and that will be carried over by somebody else. Okay? And I want to talk about few things. Maybe probably already discussed these 2 days. I'm not sure. I did not read what were the topics. But few points what they are in revolutionizing healthcare in all over the world. First one is artificial intelligence. New AI applications. What are these new AI applications? They are how they are working in medical community. Number one, virtual health. What is a virtual health assistant? Because people are very, very busy right now, and they do not even have a time to go to the diagnostic center, or do not have a time to meet doctor to know what problems going on in the body. So these virtual assistants, the virtual health assistants, can help from anywhere, any place, any app, or any kind of internet service. So you need not to go to them. They will be talking to you, and they can be assisting you. This is possible only by artificial intelligence. Next one is the machine learning. And machine learning, for a pretty long time, we do not discover any new drugs. Some drugs are coming into the market either by addition of some molecule, or addition of some salt, or addition of some kind of a new thing to that, but we have not discovered any new drugs. And hopefully this machine learning will help us to discover new drugs. Another one is robotic surgeries. Robotic surgeries also can be done. People virtually, they need not to be present here. This is all possible by artificial intelligence. That is the first point in revolutionizing. And second one is data breach prevention. What is a data breach prevention? I know a lot of PharmD students are going to the hospitals and they're talking, and you are getting the information, and you also getting the patient charts and patient information with the permission of the doctors. But that is right. With the permission we can take. But there is a possibilities in the world, there is a data breach. Means lot of people can take our information, the patient information. Now you know that cyber crimes are happening. Same thing, cyber crimes are happening to take the money or take something. Same thing, nowadays people are doing a data breach of a patient. So this is possible by the innovation in technology. Okay? And not only this, how we can prevent this data breach? First thing is the facial recognition or some kind of IT infrastructure development in the healthcare that will prevent the data breach of a patient by somebody else who are not related to us. And third one is a nanomedicine in digital thing. So nanomedicine in digital thing is what happening. This is one of the hottest topic where lot of biotech companies and lot of research is going around the world. What is this? We can have a wearable and devices and also apps which can be developed where we can have a nanomedicine in digital healthcare can be innovated and put it in a technology. Okay? Other one is social determination of health. Social determination of health gain value. And not only treating the patient, not only taking the data, not only preventing the data from stealing, not only taking care of a patient in a different way, but also social determination is also more important because the statistics and quality and affordability can be given by developing this one. Because the statistics will help you to determine how many people are having a particular disease, how many people are suffering from particular disorder, or how many people can afford that medication, how many people can need a help. This all can be done by innovating in the social determination of health gain. Another point is a smart implants emerging healthcare. Smart implants emerging healthcare. This is the hottest topic, the 3D printing. 3D printing, you know that 3D printing can be replaced with a bioengineering. And 3D printing can be helped with a whatever the injuries happen to the skin, or injuries happen even to the heart, injuries happen to even to the ear or anywhere. They can be 3D printing implants can replace, and these are biocompatible and biodegradable if required. So this is also possible by our innovation technologies. Okay? And other one is a integration of healthcare system with a big data or data silos. Okay? What is this integration of healthcare system with big data and data silos? Nothing but we have to maintain the data of a patient. Data of a patient and can be retrieved if it is required. This is all the storage devices that we can keep for years together. Means we can keep it like, even if the data from the infant to 90 years, the data can be maintained. That is also possible by this. Okay? Another one which we need a revolutionizing is the payer-provider bonds with a strengthening the patient capacity or strengthening the patient benefits. Payer-provider bonds. Payer-provider bonds is nothing but how many of you have a health insurance? How many of you have a health insurance? 1 and 2 people, 3 people, 4 people. And what that insurance by whom? By your parents, isn't it? Or is the college is providing any, or APTI is providing anything, PCI is providing anything? No. Nowadays, the PCI is going to provide the healthcare insurance. Okay? That was a new thing they have to do. provided bond or a payer provided bonds is nothing but a health insurance. Okay, in US, when they're born, when they, even before birth, the parents has to take the insurance. Okay, because if the child or the kid have any problem, that insurance has to cover from the 0 minute. Okay, so that's why they prepare ahead of time and buy the insurance for their child. They will tell, the approximate date of the birth of a child, and the insurance will be, the child insurance will be included in that. Okay, why this is important? Because this will provide the whatever the customer or a patient benefits. Because now you know that if you go to any corporate hospital, for any small treatment, you have to spend lakhs of rupees. But if you have an insurance, at least insurance will cover 80% of that. That kind of things are not happening in our country. Only few people, for us, it is a mandatory, so we take it. The people, unless it make a mandatory, the health insurance is not taken by the public. But by using this healthcare system, we can have an insurance with a payable amount every month, and that can be covered your 100 years plan of your life, 100 years span of your life. So that is one can be possible by this kind of a techniques. And other one is adoption of telehealth. Adoption of telehealth. When did we use this one telehealth? When we have a COVID time, isn't it? When we have a COVID time, that is the only device that we could call somebody to know what to do. But after COVID, how many people contacted the doctor on a phone? Zero. No, nobody will do it. They always take the appointment, sit there for 4 hours or 5 hours and see the doctor, then take the prescription, then buy the medicines. Why? How much time you are wasting? Maybe that 3-4 hours you can have some kind of work that is productive. So that's why with these innovations in AI or with innovations in internet things, we can have a healthcare by telehealth. What is telehealth is nothing but we take an appointment either by Wi-Fi or by any kind of internet service or by email or by any kind of video conference or streaming services, even Zoom meeting. That will only take about 5 to 10 minutes, and you will be saving your time for a transport and waiting for your turn to see the doctor. So this is also possible in this era. Another one is virtual realities, augmented realities, and mixed health realities in healthcare. This is one of the most important things to make decisions in a healthcare. This virtual, augmented, and also mixed realities. All these are one of the most important things which can make any kind of a decision required for us to proceed in a healthcare. Another one, a most important one, overcoming the mental health challenges with the telepsychiatry. Overcoming mental health challenges with telepsychiatry. What is this? And you see that lot of people in the paper, or lot of them in the TV. Somebody has did the suicide. Somebody has jumped from the building. Somebody was, feeling loneliness. Somebody was feeling like, they are isolated, and also they feel like they might be having a chronic stress, or somebody might have burnout, and some kind of anxiety, some kind of a depression, some kind of emotional fatigue, some kind of social isolation. All these will lead to the mental health. These mental health might be some reason, some kind of a problem. Some might be stressed out with something, maybe stressed out with exams, stressed out with some kind of problems, or stressed out with some friends, or nowadays boyfriend, girlfriend problems. So many are there. So Emotional stress and mental health is the one of the things that can be taken care by innovating technologies. Why I am taking? There is a people, you can talk to them. There is a people who can discuss your problem. Need not to be publicized, and you can talk to lot of good healthcare people, and they motivate you to come out from that kind of a stress. That is all possible. And never ever feel a mental health. Never ever feel a stress. And whenever you have a stress, try to be sit down and relax and count 1 to 100 and breathe heavily for 5 minutes or do some kind of a meditation. And that is all will solve by itself. But still it is not possible. Take a help from the healthcare, which you don't have to go out of the house, and there are people who can help by one of these technologies. Okay? Another one, nowadays more important, that 5G enabled devices in medicine. That we have to expect that one very soon. 5G enabled devices in the medicine, or apps, or any kind of devices. This will give you the correlation between the demographics and ecology and also economy, and information is also available, and patient data can be fed to that, and AI and ML algorithms will analyze and give you interpretation and give you the actual health, whatever the health care required, that is also can be analyzed, and with a low cost it can be given. That is the big data, that is the 5G enabled devices and apps. These are all will help you when you feed the data of a patient. There is a problem, diagnosis and disease, the medicines what they are using. They are available, they will be analyzed in a whatever the way you want. If you are in India, it has to be analyzed in US and Canada. That also can be done, and they can give you the suggestions to take care. Another one is big data analytics. These big data analytics are nothing but these are the data collected from all over the world, and for one disease, how the patients are feeling in all over the world is given to the big data. This big data will give you, whenever you want to analyze that kind of a disease, this big data will give you how many people are suffering from there, what are their symptoms, what are your symptoms will match, and what are the medications that they are taking, what are the medications you can try, that will be given. Means everything in the world, probably millions of people might be having same problem. That data is available with one click, and you cannot feel that you are the only one suffering from that, but there are many people are suffering. And what they are doing, what kind of analysis is given, what type of medicines can be done, and this you can learn, and that can be implemented in your life. Another one is also a gene therapy. Gene therapy is nothing but study of a human with a gene difference. So each one has some difference, because we are born here because we have to do something. We are born here, there is something that you have in the life to do it. Okay? That's why you are born here. So same thing, the gene is also there, that is specific to you only, specific to me only. It never matches with any other person. So these genes can be analyzed, and these genes can be give you to give a precision medicine or a personalized medicine to you, or personalized medicine to me. Means they will make the medicine which is only useful for you by studying the gene therapy. That is also possible. And same thing, this with the gene therapy, we can give a precision medicine. So precision medicine means that if you take a drug, it will be not only acting on that organ, it will be acting throughout the body, and the same medicine has to be used by many people. It will be one kind of reaction to me, one kind of reaction to you. But when you do the gene therapy, by analyzing that, you can have a personalized medicine to you that will be acting on you, and that will be acting on your target. So these are all kind of innovations can be possible by revolutionizing the healthcare through the innovation in technology. And this is some kind of a revolution in the healthcare. It is not only for us, it is all for the pharmacy and medicine. And try to be doing something in your life and prove yourself that you can do something, and that might be helping the future kids. Okay? And thank you for this opportunity. And hope in future I might learn, or one of the MLR students or the people who are attending this conference will develop one new drug or many new drugs which can be helpful either through the artificial intelligence or machine learning or big data, augmented, anything, any kind of a healthcare that you can adopt, and we might see you in the paper and news in a big way. And I wish you all the best.
We express our sincere gratitude to Dr. Sunita M. Reddy Garu, Professor and Principal, JNTU University College of Pharmaceutical Sciences, Sultanpur, for addressing the gathering with her insightful and thought-provoking remarks. Her gracious presence and valuable message have greatly enriched the valedictory section of this conference. Thank you, ma'am.
Request Dr. Malik, Dr. Mahendran Sekhar Garu, doctorates to felicitate Dr. Sunita ma'am. Good evening. Now it's time to make you guys more happy. I congratulate all the winners for today. First, the winners of oral presentation. It is special appreciation prize to Anusha Tejasri Manogna from Guru Nanak College of Pharmacy for presenting automation and robotics in pharmacy.
Maybe you should start learning math now.
Next special appreciation prize to Nikita from Marri Reddy College of Pharmacy. Next appreciation prize to Indrani, Naipur, Hyderabad. Next is third prize for Krupa and Jyotirmai from Guru Nanak College of Pharmacy, drug delivery system. Next third prize, Lata Shri, Marri Laxman Reddy College of Pharmacy, AI driven pharmacy, the game changer in modern healthcare. Second prize, C. Swati, Marri Laxman Reddy College of Pharmacy, Digital Twins in Healthcare, Revolutionizing with the Living of Heart Project.
Second prize, Niharika, CMR College of Pharmacy, Beauty Parlor Stroke Silk. First prize, Shivasai Venkata Karthik, Gokaraju Rangaraju College of Pharmacy. Next are the winners for poster presentation. First prize, Shivasai Venkata Karthik, Gokaraju Rangaraju College of Pharmacy. Special appreciation for poster presentation, Sri Ganga BITS, releasing photothermal coating for accelerated infected diabetic wound healing. Third prize, B. Pavan Kiran, Gokaraju Rangaraju College of Pharmacy, types of nanosperm sensors used for detecting of cancer cells. Third prize, A. Kovida, CMR College of Pharmacy, molecules from imagination, AI-guided Nova drug design using generative models. Second prize, Mankala Lokesh Kumar, Marri Laxman Reddy College of Pharmacy, smart packing of drugs, enhancing patient compliance and drug safety. Second prize, Rahul Udayraj, Motishri College of Pharmacy, AI biochip disease detection in early stage. Second prize for Rahul Udayraj for AI biochip disease detection in early stage. First prize, V. Sai G. Vinay Kumar Reddy, Tigala Ram Reddy College of Pharmacy. Emerging technologies like AI in clinical pharmacy, personalized medicine, and addressing challenges in medication management. There are 2 more special prizes for poster. Lakshmi Prasanna, Vijay College of Pharmacy, Phytosomes in Novel Drug Delivery System. Next is prize, U. Vaishnavi, Bochan Nasimlu College of Pharmacy, Pharmacists Meeting the Global Health Needs. Is anyone there to take a prize? Bochan Nasimlu. Thank you. I now request Mrs. Anjana Saraswati, Assistant Professor of Department of Pharmacology, to propose a vote of thanks.
Good afternoon, everyone. Good afternoon, everyone. We are closing the session now. I want to hear. Good afternoon. As we conclude this intellectually invigorating and inspiring 2-day journey, it is my honor and privilege to welcome you all to the valedictory ceremony of the International Conference on Pharmacy 4.0, Revolutionizing Healthcare Through Innovation and Technology, organized by Marri Laxman Reddy Institute of Pharmacy in collaboration with Telangana State Branch of APTI. This conference has been a remarkable platform for knowledge sharing, collaboration and forward-thinking discussions on the transformation of pharmaceutical sciences in digital era. We truly are delighted and deeply honored to have amidst us our chief guest for the valedictory session, Dr. M. Sunita Reddy Ma'am, Professor and Principal, JNTUH University College of Pharmaceutical Sciences, Sultanpur. Ma'am, your presence has been a source of great inspiration to all of us, with your immense contributions to pharmaceutical education and research and your unwavering commitment to the academic excellence. Your words have surely left us a lasting impact on everyone present here. Throughout this conference, we have had the privilege of hearing from our distinguished academicians, researchers and industry leaders who have shared transformative insights from digital health and manufacturing to data-driven drug discovery and patient-centered innovation. Let me take a moment to share a small anecdote that truly captured the spirit of this event. On the very first morning, I overheard an engineering student remark saying, "I thought pharmacy was all about pills and prescriptions. But today I realized it's also about coding data and even robots." And this was actually said by one of our engineering students from MLRITM. So that one statement encapsulated our goal to open minds, shift perspectives and inspire innovation in the world of pharmacy. I extend my heartfelt gratitude to our visionary management, Shri Marri Laxman Reddy Garu, Honorable MLA and Founder Secretary, Shri Marri Laxman Reddy Garu, Chairman, Srimati Mamta Reddy Garu, Treasurer, Ms. Anushriya Reddy Garu, Secretary, Shri Marri Dhiren Reddy Garu, our Director. Your unwavering support and constant encouragement have been driving the force behind our success of this conference. A special note of appreciation to our convener, Principal Dr. Arnava Malde Garu, co-conveners Dr. Surendu Saha and Dr. Hukain Mabendu, Organizing Secretary Dr. Saidat Das. Thank you for your vision, dedication and tireless efforts in organizing this event to perfection. I also express my sincere thanks to our faculty coordinators, Dr. Gopi Krishna Garu, Dr. Harita Ma'am, Organizing Committee and our enthusiastic students. A big applause who have worked
the least, I thank all the delegates, presenters and participants. Without your active involvement, thoughtful presentations and valuable discussions, we wouldn't have made this event a grand success. Thank you. And this is Anjana, Assistant Professor MLRIT, and it's been a very privilege to propose the vote of thanks on behalf of the organizing team. Thank you once again. We wish you all a continued success and meaningful collaborations in the future. Thank you. And last but not the least, again, thank you, Vyula Rani ma'am from TRR College for being a constant support in getting number of registrations. A big shout out to Vyula ma'am. And lastly, we have our sponsors who are not present here, but yet it is our responsibility to thank them. Are the Overseas Consultancy, Mr. Srikanth, and the Uni Expert, Pawan, who has been constant support throughout the system. Thank you. I request all of you all to please stand up for the national anthem.
भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
All the prize winners are requested to come near the stage. We will have a picture with our guest panelists. Panelists also please join us for a picture. Students, get your pictures clicked by our cameraman. We will share all those pictures via email to your faculty. Please come and join us for a picture.
You are patient.